pleasure for me. Can you hear me? Not so good. Now it's okay? Thank you. So it's a great pleasure for me to start this 15 great meeting. I mean, it's amazing that we are still here after so many years, still continuing this adventure. And I'm so happy to see that there are a lot of people that, regardless of many difficulties, are coming from their own country, from Europe, from Asia, from South America. This is something, and from US, of course, and this is something that makes our job really concretely approaching a new results. That is that we are contributing in research. It has been a very difficult time for all of us involved in every day fighting with this terrible pandemic, but nevertheless we are continuing to make this spirit of research, and still we are publishing many papers, and the young people that are really the basic of this group is still working regardless of the difficulties. So it's my privilege to say welcome to you in Italy. Welcome to you to the beautiful city of Florence. It's not Rome, but it's still beautiful. And I think that would be good to have good science, but also good time together, like the Spirit of Great always does. And also don't forget that we have a special event on Saturday, actually two special events. The first one will be the soccer event, that this time the group from Christian and Alex will try to win with Italy. I'm not sure they will success, but also the fantastic opportunity to visit the Uffizi. So again, thank you for coming, welcome, and I really appreciate your effort and hope there will be a good time for science and to have fun together. That is the spirit of Great Network. Thank you.
Okay, nothing else to add except that indeed the Italian team is going to lose this year in soccer. Thank you for coming. It's really a great opportunity because we manage, I think, during these COVID years to be still present and have still collaborations, publications, having the young generation on stage. And it's really even a dream come true that we are all together again this year. The program is really incredible. There are really many data that we are going to see, and the spirit of great is still here, meaning that really the old generation will be just asleep while the young generation will show a beautiful data. And please don't hesitate during the break time, during the dinner, to match different teams, to try to have new ideas, share data, because this is really the spirit of Great came because we thought that we need to have the young generation collaborating and sharing data and having common publications. Then again, enjoy this meeting, and you will have really incredible presentations that you will listen to.
It's my pleasure. So it's my pleasure now to highlight to you some specifics of the program this year, and again, perhaps one specific detail of the Great Meeting in general. I want to highlight that the Great Network is not only research collaboration among academic colleagues, but it's also a wonderful research collaboration between academics and researchers in the diagnostic industry. This is something very special, and therefore this character is also present at the Great Meeting. So I want to highlight the important role that the representatives of several companies have had over the last years to maintain and to develop the meeting as it is, and also to have a very active role during this meeting, as you will see in the program. The first session, we will have Tobias, Cecilia, and Niels on stage, and they will share the latest news that is derived from their technology. You can also see that this year, for the first time, we have 2 awards. It was quite difficult to convince my colleagues that yes, we need the money for 2 awards, but finally they agreed, because I think a competition is nice, but a competition must be fair. And in order to have a fair competition, we decided we need 2 categories. One category where we have the youngsters on stage, young experts on stage. These are usually researchers who are at the beginning of their academic career and have the benefit, of course, using the Great Network as their first international appearance. We are very happy to also have more senior colleagues, and they have presented at several meetings before. Many of them already have won a prize at these meetings, and so they, of course, should compete in their own category. And therefore, the second category is called senior scientists lectures. The award committee is completely independent of the 3 of us. And I'm very thankful for the colleagues who have agreed to work on the committee due to the illness of some. We had some changes, and I'd like to thank Frank Peacock for, first of all, having been here for so many great meetings and having traveled from the US and really helped to that this is really not only an international, but really a global meeting. So thank you, Frank, very much for your efforts. And Frank agreed to chair the award committee this year. He is supported by 2 colleagues that many of you know. I haven't seen Josep. Has he arrived? No, but he will hopefully in the next minute. So Josep Masip from Barcelona and Shaw Collins from the US. And he is supported by 2 lady scientists, and I think that's a particularly important achievement. And in fact, 2 lady scientists who had already attended as youngsters on stage the first Great Network meeting 14 years ago. So they were present in Rome 14 years ago when we had our first meeting, and Nisha Arinya and Theodore Socrates, they will also join the committee. So, having said that, perhaps a last sentence just on COVID. I think if you go on the train in Italy, you have to wear the masks. In many of the countries, COVID is on the rise, and so I think from our perspective, of course, I think the legal point of view, we do not have to wear masks in the room. But as you realize, some of the colleagues in the room prefer to wear a mask. I think this is smart. This is 100% appropriate. And I think everybody should either wear a mask or not, how he or she feels comfortable. I think that's the part at this point in time. It's not obligatory, but everybody should either wear a mask or not, however he or she feels comfortable. Good. I think having said that, I would like to start the 3rd session. Is that okay?
Yes.
Perfect. So with great pleasure, I would like to ask Tobias Becher to come to the podium. Tobias is an expert working in Roche. He has a background as a basic scientist. He has worked as a clinician, so he knows medicine from very different angles. And Tobias, we are very much looking forward to your presentation on clinical decision support in heart failure, from raw data to actionable insights. Thank you, Tobias.
Thank you, Christian, for the nice words in the introduction. Well, then I say thanks again, Christian, for the kind words in the introduction. And then I have a pointer. Perfectly. Well, thanks a lot. Good afternoon, everyone. My name is Tobias Becher. I'm a clinical development lead at Roche Diagnostics International in Switzerland. And it's my great pleasure today to talk about clinical decision support in heart failure from raw data to actionable insights. As you will realize soon, this is also a project for us that is work in progress, so we're kind of collecting input and insights right now. And what I want to share with you today is our vision around what we're trying to achieve within the next years to move this field forward. So briefly, today I'll talk a bit about telemedicine and remote patient management. I want to dive a bit into the current evidence that we have for remote patient management, the challenges and the hurdles, and then again share our vision of what we try to achieve as a company within the next years. So briefly, telemedicine and telecare is defined as diagnostics and therapy that use information and communication technologies. And this can be aimed towards supporting the interaction between healthcare professionals and providers on the one hand, but also can be aimed at supporting the interaction between healthcare providers and patients. I think we kind of lost the presentation. All right, thanks. So under the segment of remote patient management, you can see there are a variety of different modalities that can be counted towards that. One is teleconsultations, so the use of virtual visits and virtual follow-ups. Disease management services in home care, such as virtual care plans that are adapted towards the individual patient needs. And lastly, also remote patient monitoring. And the bucket of remote patient monitoring is quite diverse. You see here, non-invasive measurement of vital parameters can be counted towards that. The use of biomarker measurements at home, such as blood glucose and others, or the monitoring using implantable devices such as PA pressure monitoring. So along heart failure patient journey, there are multiple opportunities to use remote patient management tools. So on one hand, we can try to increase or enhance the diagnosis of heart failure. We can identify high-risk patients and try to tailor the intensity of the management. We can try to tailor therapy and adapt dosing along guideline-directed medical therapy. We can monitor patients and identify patients that are deteriorating early and try to intervene to improve outcomes. And lastly, of course, we can try to improve outcomes overall, so reduce length of stay and decrease mortality. So there are multiple benefits so far that have been postulated for remote patient management. On the one hand, we can aim to increase access, so patients that are not usually go to their physician or live in remote areas. We can support patient empowerment, so help patients take care of their own patient journey and empower them to take their own decisions, informed with the help of a physician. We can try to improve outcomes, length of stay, readmission rates, mortality. We can try to integrate care across the patient journey, so try to connect different specialties that are involved in heart failure patient care. And lastly, we can also try to save costs along the patient journey. So what we've also noticed when developing these solutions is that there is a number of challenges along the way. So it's not as if you are implementing improvement in medical therapy or diagnostics. Here you have to overcome significant hurdles with healthcare providers, such as lack of awareness, difficulties with adoption into the clinical workflow, and lack of evidence. And on the patient side, you also have to face IT literacy and access. But beyond that, there are a number of other challenges, such as with payers, lack of reimbursement in a lot of countries right now, fragmented legal framework and concerns with data protection and privacy. And lastly, also IT challenges where you have to match different infrastructure and resource needs, and you have to provide interoperability between different solutions. I think what we've seen during the last 2 years is that COVID-19 was one of the significant drivers for the adoption of remote patient management, and specifically here, the increase of numbers of remote visits. What you can see here on the left is data from Israel. On the top, you see the number of daily COVID-19 cases recorded, and in gray, a period of lockdown. And you can see that during this period, the number of remote visits increased significantly, and more importantly, after the lockdown, it kind of stayed increased compared to baseline, showing that there was some adoption and some benefits drawn from this kind of patient care that was significant enough to be maintained. And the other thing that you can embrace here on the left side is that we've also expanded into different areas of the patient journey. I would argue that before COVID, we mostly used remote patient monitoring in this home monitoring space, while after COVID or in the post-COVID area, we're using it in areas such as telerehabilitation, virtual visits, and forward triage. So now the question is, what kind of evidence do we have? And I just want to provide some of the evidence that I found. So this is basically highlighted in the current ESC heart failure guidelines. This is a meta-analysis from Lin et al. That looked at 39 studies that looked at telemedicine as an intervention, so teletransmissions or telephone-supported care, and looked at the impact on outcomes such as all-cause mortality and heart failure-related mortality, length of stay, and mortality. And the conclusion from this study was that overall telemedicine was shown to be beneficial, with home-based teletransmission effectively reducing mortality, admissions, length of stay, and mortality in patients with heart failure. The evidence is a bit different when it comes to remote patient monitoring, and here it really depends on what technique you're using. So if you look at non-invasive measurements such as weight and home measurements, you see that studies were mostly neutral, except of TIMI-HF. Same is true for thoracic impedance measurements and multi-parameters, where you try to merge different tools into one score. The exception is hemodynamics, so invasive measurements such as LA pressure measurements and PA pressure measurements. And this is also reflected in the current guidelines. So here from the current ESC heart failure guidelines, you see that non-invasive home telemonitoring may be considered. So it's a class B level of recommendation with a B level of evidence. And the same is true for pulmonary artery pressure measuring to guide patient management. And for ACC, I would argue that here it's even more cautious. So here they assign an uncertain value, same level of recommendation, but here the statement is it's an uncertain value. So what do we need to change in order to make remote patient management more feasible and really to drive adoption? And this is the part where I would like to share the vision that we have at Roche to accomplish that. So we believe that a final product or a solution that we're providing should be an integrated solution. And we are working towards developing, on the one hand, evidence-based disease management, so care pathways that are delivered across the IT platform and that support evidence-based care protocols on one hand, and try to merge it with different areas of remote patient monitoring. And we believe that although this may come towards a solution, there's also work that needs to be done in terms of adopting this, and this is where we think that healthcare consulting or adoption management could play a role. So here we are currently looking into data analytics and change management, and also patient pathway optimization, because we've seen from a lot of studies that although you may show a benefit of a tool, the adoption part is also equally important, and sometimes it's really difficult to tease apart what change was driven by your product versus what was driven by the adoption of that. And the final vision would be to have one, an integrated patient interface that aims at empowering patients, and two, a dashboard that could enhance patient-physician communication and support the physician in taking decisions and integrating data into something meaningful. And hopefully this would lead to a continuous interaction between the physician and the patient on one hand, and on the other hand, it would help to manage a patient, to manage their own disease with supervision by the physician. Lastly, what kind of projects are we currently conducting to drive this forward? So one development that you see here, and sorry for the problems with the formatting, is a clinical decision support platform, which is a post-acute care management solution that is built on a platform which would be integrated in the EHR and shows a comprehensive overview of the patient journey, the encounters with the physician, and the data that was collected along these encounters. And here we would use that data to provide care recommendations and algorithms along the patient journey. On the other hand, we are currently conducting pilot studies to look at different ways of remote patient monitoring and how to integrate that into this solution. So this is looking at supervised patient self-management, embedded care protocols, and medication optimization. And lastly, we are, together with Kari Lam, looking at combining biomarker measurements such as proBNP with point-of-care echocardiography that is supported by AI to improve diagnostics in the heart failure field. And then at the end, hopefully come towards an integrated solution that can be applied across the patient journey. So as a conclusion, we believe that telemedicine has the potential to transform healthcare in the heart failure space as we know it. We've seen during COVID-19 that the potential is there and that there's ways to adopt these solutions that actually lead to a benefit. However, to really drive that forward, we will need an integrated evidence generation and also measures to address different stakeholders and their concerns. And lastly, our future developments will aim towards developing an integrated solution that can be used across the patient journey. Yeah, that's it from my side, and I hope with that I've inspired some questions, so please feel free to ask any of them.
Thank you, Tobias, for this fantastic opening of our Congress and highlighting kind of the new options in clinical decision support in heart failure. Please raise your hand if you have a question, and you will provide a microphone. Please. Do we have a microphone for? Yeah. Yes, thank you.
Can I take the opportunity of the microphone?
Yes.
Congratulations, it was a really fantastic presentation. I think you really opened a new door for the future of management of patients that we see in the emergency room with acute heart failure with the different phenotypes, right? We have the very critical patient that we should admit for ICU, but then we have a lot of patients that we just need to adjust a little bit the treatment. And then one question is, I have two questions. The first one is, do you need a nurse's dashboard management to telemonitoring patients? So the data are coming from the home to a nurse's station and then to the doctors. This is one question. And the second question, you mentioned the most important thing to monitoring natriuretic peptides to be this fantastic opportunity to control and predict the patients that are worsening and also prevent hospitalization. So in the second question, should you tell us how is this technology? Is it point of care? How does it work?
Okay. So firstly, I think towards your first question, that is something we're looking at is what is the best way for a physician to access the data and integrate it into the current workflow? And I don't think there is a simple solution. I think what we've learned is that we should not add additional burden to the physician. So usually physicians are very reluctant to use another app, another program that pops up, something that adds complexity to the workflow. So I think the general thinking is we need to integrate it into the EHR system. And that can be difficult depending on what country you are. We are currently working a lot in the US because here you have a market with EMRs that is a bit more focused towards Epic and Cerner, so main providers, while in Europe it's a bit more fragmented. And here I think the IT solution itself and the development of that is a major hurdle. But at the end of the day, I think the goal would be to create something which is as easily adaptable as possible to the workflow. And I think towards your second question, with Echo AI, we're currently exploring with Carrie Lam the opportunities of using echocardiography at the point of care, so training nurses to use echocardiography devices and artificial intelligence to obtain images and also to interpret them at the bedside. And here it needs to be seen if NT-proBNP can augment that either measured by a GP or if there's really the opportunity to develop a point of care device.
Richard.
Thank you very much. That was very interesting. The question I have is in our system now, we are developing the home hospital concept. So when somebody comes into the ER, as Sal said, who has heart failure or COPD, and they're older or you're a little worried, do they really need to be in a hospital at great expense? And so the pilot program is to actually, those people that are sort of on the edge, you send them home. And what happens is a nurse visits them 2 or 3 times a day. The physician rounds on them in the morning. And if they feel they're getting worse, they can get directly admitted back to the hospital. And what's really interesting is that it seems to be working, and actually patients like it because they eat their own food and things of this nature. And I think it's primarily driven by cost. And so the original biomarkers would all be done in the ED, and you could repeat them, and the nurse could take them or whatever. But I think a lot of this is driven by cost. And I think when I think about it in my career out of the ER, there's many patients that were on the margin, but we thought, oh, we should admit them. And the question is, especially in heart failure or COPD, do you really need to? I mean, what happens? They sit on a ward, they get seen, 2 or 3 times a day, and of what benefit is that really? So I think this movement, however it goes, will continue, and maybe the hospital will one day become one big ICU.
Yeah, I mean, that's, at the end of the day, I think cost reduction is one of the strongest drivers that you can have, especially in the US. So even sometimes before evidence, cost reduction itself is enough to drive adoption into a system. However, we would also like to see that there is actually improvement in some of the things that we do to patients. And I agree with you also from my experience as a physician is it's not to the best effort of a patient to be on a ward for a couple of days just to be monitored and actually nothing is done. It doesn't really help the patient. So moving this actually the point of care outside of the hospital and still making sure that you can maintain the quality of your care is, I think, something that we should try to obtain with those solutions.
Okay, I think if there are no other questions, oops, thank you very much, Tobias.
Yeah, thank you very much.
Now is for me a pleasure to introduce Dr. Cecilia Scarponi from Orthoclinical Diagnostics. She's going to talk about the role of IL-6 in cardiovascular disease management. Cecilia.
Thank you. Perfetto. Here we are. Good afternoon, everyone. My name is Cecilia Scarponi, and I'm a part of the EMEA marketing team, scientific team at Orthoclinical Diagnostics. As introduced, I will speak about the role of IL-6 in cardiovascular disease management. So, IL-6 was identified in 1973 as a soluble factor that is secreted by T cells as important for antibody production by B cells. Today, we know that IL-6 is a pleiotropic cytokine with a wide range of biological activities in immune regulation, hematopoiesis, inflammation, and oncogenesis. Since nearly 50 years now, it's clear that IL-6 pathway has emerged as a pivotal pathway involved in immune regulation. And more recently, severe COVID-19 may be regarded as a viral-induced hyperinflammatory condition with multi-organ involvement due to a cytokine cascade. Soon after the beginning of the COVID pandemic, IL-6 elevation was identified as risk predictor for disease severity. And this explains why, in 2021, the WHO has updated its patient care in guidelines to include IL-6 receptor blockers in addition to corticosteroids in case of severe or critical COVID-19. And IL-6 is still commonly used in hospitalized patients to predict risk of respiratory deterioration. So targeting of the IL-6 pathway has led to innovative therapeutic approaches in multiple diseases. Here, there's a table which shows an overview of interleukin-6 clinical applications. And as you can see, there are many different cases where this is really important. So for example, in rheumatoid arthritis, the IL-6 pathway inhibition is effective in many clinical trials, has shown to be effective in many clinical trials, but as well in cytokine release syndrome, tocilizumab, which is an interleukin-6 receptor blocker, was used to successfully treat CRS, occurring specifically in trials of CAR T-cell therapy. So here, this is an overview of the many different clinical applications that interleukin-6 may have. So a brief regard now going to heart failure. So heart failure, as you all know, is affecting over 40 million people globally, and it's the most frequent cause of hospitalization in patients over the age of 65. Overall, the incidence is increasing due to aging of the population, and typically 50% of heart failure patients have heart failure with reduced ejection fraction, the remaining 50% accounting for preserved or mid-range ejection fraction. In the United States, adults with any diagnosis of HF have a 5-year survival rate of roughly 50%, and this is pointing at the fact that we really require tools for early recognition and to initiate goal-directed therapies. Studies have shown already the association between elevated inflammatory markers like CRP, interleukin-6, and tumor necrosis alpha with the prognosis of heart failure. So let me speak here about a study appeared and authored by Albar et al. That really wanted to establish these inflammatory markers and the risk of heart failure with a reduced or preserved ejection fraction. So the study aimed at determining this association of interleukin-6, interleukin-2, tumor necrosis factor alpha, and CRP, but also particularly pointing at the different subtypes. Follow-up period was 10.9 years. So let's see the results. So first of all, it's important to notice that the results show that interleukin-6, tumor necrosis factor alpha, and CRP were associated with all heart failure subtypes. However, interestingly, only IL-6 and CRP were associated with incident heart failure with preserved ejection fraction. And only IL-6 remained statistically significant in Model 3. Model 3 is controlled for all other biomarkers. So this with a hazard ratio of 1.78. So this is really pointing at the fact that the authors concluded that IL-6 and CRP are associated with incident heart failure with preserved ejection fraction, but not with reduced and mid-range ejection factor. And this suggests that activation of the IL-6-CRP pathway as a cause, consequence, or epiphenomenon may be unique to heart failure with preserved ejection fraction. Let me show you here a fantastic review that has been appeared in, published in JACC in 2020 and authored by Murphy and Januzzi. So here on the right, we can see the organ crosstalk with the heart in the middle. So indeed, the heart is speaking with all the other organs, and there is this inflammatory situation which is extremely complex because some factors do not only originate from the heart itself, but also trigger other complex mechanisms that are still quite a lot to be understood. So it's clear that IL-6 contributes to the pathogenesis of comorbidities coexisting with an exacerbated heart failure severity. Observational studies found correlation of IL-6 concentrations in plasma with worsening HF and renal dysfunction. And there is a retrospective study, Biostat CHF study, that is investigating the relationship between IL-6 and the clinical characteristics, outcomes, and other biomarkers. The association was found with atrial fibrillation, lower eGFR, higher NT-proBNP concentration, abnormal iron metabolism, and poorer exercise tolerance, so leading also to anemia. Elevation of IL-6 concentrations were associated, again, with heart failure with preserved ejection fraction, and each doubling of IL-6, this is interesting, predicted HF hospitalization, cardiovascular, and all-cause mortality. So the authors point at different areas of studies in order to further understand this and point specifically at the fact that defining a specific subphenotype of HF with respect to these anti-inflammatory therapeutic strategies may be useful, focusing particularly on a subphenotype, which would be heart failure with preserved ejection fraction with chronic kidney disease, iron deficiency, and atrial fibrillation. So in reality, it's really still to be understood whether inflammation is causing heart failure or heart failure is, so really the mechanism. So let's now have a look at the relation between IL-6 and STEMI. This is a study by Tiller et al. Published in 2022 this year and investigated the association of IL-6 concentration with infarct size, reperfusion injury, seen as microvascular obstruction and intramyocardial hemorrhage assessed with CMR, adverse left ventricular remodeling, and major adverse cardiovascular event. The population study consisted of 170 consecutive STEMI patients, and inclusion criteria included first-time STEMI, revascularization by primary PCI with 24 hours after symptom onset, and EGFR more than 30 ml per minute, and KILIP class less than 3 at time of CMR scan. Yeah, I forgot to say that IL-6 was measured at day 2 after the primary percutaneous cardiovascular intervention. So what do the results show? Well, the results show that in all the patients with IL-6 concentration higher than the median of 17 nanogram per liter, there was a lower left ventricular ejection fraction, a larger infarct size, larger MVO, and more frequent IMH, and also more common LVR. In addition, in order to see how the clinical outcome, whether measuring IL-6 had a clinical outcome, it was associated indeed, higher IL-6 values were associated with a lower major adverse cardiac event-free survival rate. And in conclusion, the authors concluded that the role of IL-6 can be seen as a prognostic biomarker for more serious outcomes in the early phase after STEMI. Indeed, IL-6, they have seen that improved risk stratification after STEMI, and its role in this complex inflammatory response can be a double-edged sword, in the sense that on one side, IL-6 is protecting against oxidative stress, and on the other side, signaling may lead, as we have seen in the previous study, may lead to hypertrophy and depressed cardiac function. So there is a potential role of IL-6 as therapeutic target, but in order to find clinical cardioprotection trials, like the SAIL-MI trial, for example, but still a lot of data are required in order to really understand on which subtype to focus. And I would like to conclude, to go to conclusion, to say that within the acute care comprehensive panel, Ortho do have an IL-6 test, which is traceable to the first WHO international standard and extremely well correlated with Alexis test with a correlation coefficient of 0.998. So in summary, interleukin-6, we have seen, is a central hub in cardiometabolic signaling. Inflammation can be both a cause and a consequence of our failure. The relationship between our failure and inflammation is therefore only partially understood, which make things interesting for you all. Studies have demonstrated so far a statistically significant association between IL-6 and HFpEF. This association was found with IL-6 atrial fibrillation, as well as lower EGFR, higher anti-proBNP, abnormal iron metabolism and anemia, leading to identification of specific subphenotypes of our failure with respect to anti-inflammatory therapeutic strategies. IL-6 can be used for early identification of HFpEF as a predictor for hospitalization, so predictor of outcome, cardiovascular and all-cause mortality, so as a prognostic factor. IL-6 has demonstrated its use as a prognostic biomarker for more serious outcomes in the early phase of STEMI, so it can improve risk stratification after STEMI. And finally, IL-6 has revealed its potential role as a therapeutic target for clinical cardioprotection trials, like the trial I mentioned, and more research is needed to better understand the role of IL-6 and the potential therapies targeting, indeed, both HFpEF and AMI. Thank you for listening.
Thank you very much, Dr. Scarponi. This presentation perfectly reflects the innovation spirit of the great. And I think it is really so good to think about the possibility in acute setting to measure biomarkers that is reflecting the morphology of the heart and therapeutic implication. Now, this interesting presentation is open for discussion. Dr. Mebazaa?
Yeah, thank you very much for this presentation. It reminds many of the young guys that the IL-6 is so important. Are we ready, like Salvatore was saying, are we ready to measure IL-6 like as a point of care in emergency room to try to stratify as early as possible and try to think on new therapies?
You mean in terms of as a point of care system? Well, now with Quidel, merging with Quidel could be possible, but for sure, I think it's a way to go. Sure, to make things extremely easy. Now, it is true that with on vitros, having this platform is quite rapid, so it's a short turnaround time test. But yes, I think it could be a good addition for the many reasons that we have seen to include it in an acute care portfolio. Indeed, it's a good suggestion.
Thank you. Other questions from the floor? Yes, Professor Brenner.
Do you know the source of the IL-6 that you're measuring? Is it from the heart itself or is it from somewhere else? And also, does the IL-6 levels in the serum reflect IL-6 levels in the heart?
This is a very good question. So, well, it is. This I don't know. I don't know exactly whether. I think it reflects indeed, but the origin precisely of the molecule. Yeah, I think it's in blood, in reality, but this is a valid one. I think the studies are there. I'm not aware of, indeed, to see this relationship.
Other questions from the floor?
So can you perhaps tell us the amount of interleukin-6 that is in blood? So is it an analyte that is very difficult to measure, that has very low concentration, and where kind of it's challenging in the normal range, as we have found out with high sensitivity troponin? Or is it an analyte that is more easy to measure? So do you think there are differences between different assays that might have clinical implications, or do you think it's an analyte where perhaps the analytical differences might have less of a role?
Yes, thank you for this question. Well, I honestly wouldn't compare this to HS troponin, indeed. I think there is more, it's not standardized yet, but still it's not as critical to detect extremely small concentrations as it's the case for HS troponin, where indeed the limit of detection is crucial, and we cannot compare results from one test to the other. So it's not as challenging on the analytical standpoint, and therefore I wouldn't point at a specific criticalities there. Yeah.
Okay, great. I mean, this is a really important topic, and we have data that Luca Crisialdi will present. Just looking as it present IL-6, also high-sensitivity troponin, so we'll be really open a new door that we really will look at in the future, also having this echocardiographic intelligence assessment evaluation to do immediately. Thank you, Cecilia, again.
Thank you.
Now I'd like to ask Dr. Niels Kramer to come to the podium, and it's now time to first mention the word troponin in the talk. It has been 2 talks with very little of my preferred biomarker. Niels, thank you very much for now putting this center stage. High sensitivity cardiac troponin I, central lab or point of care. The choice is yours.
Thank you very much for
Let's
paving
be
the way.
critical. Is it really time for point of care? So we are listening carefully.
Let's discuss indeed. Yeah, so my name is Niels Kramer. I'm the clinical marketing lead for cardiac point of care. And I'm very excited to present to you a group of leaders and hopefully future leaders. Maybe good to note that I have a history with this product. I used to work at Philips, where this system actually is developed. I worked there for 2 years and 6 months. Then the business was closed down. I now work 2 years and 5 months at Siemens, so I don't know what the future holds for us. That being said, currently at Siemens we have the possibility to offer high sensitivity troponin I at the point of care or in the central lab. And I think it depends on your setting and on your preferences and how your workflow is set up, which solution is preferred and which suits your setting best. One of the potential settings as depicted in the slide that could benefit from the point of care based solution is the ambulance. But apart from that, we also think that there is a need for a rapid diagnosis in the emergency department. As you know, there's a universal problem of crowding in the emergency department. Chest pain patients are the second largest group of percenters, but only 1 in 10 actually is experiencing a heart attack. So that means that, and even moreover, 50% or even more is not experiencing something cardiac related at all. So that means that for a large portion of patients, you potentially can rule them out very quickly. And that's also where we believe that a Teleca VTLI or cardiac point of care solution might play a role. So what we offer is a high sensitivity troponin result within 8 minutes. And we believe that that potentially enables the physician to make a decision within one patient interaction. So the system consists of the following components. We have a handheld analyzer with Wi-Fi capabilities. There is a docking station. The docking station itself can be used to charge the system and also can be used to have a cabled internet connection. Then there's the consumable, the cartridge, which can be used with plasma, venous whole blood, and also capillary samples. And we obviously have the ability to hook up to a data manager. So the interesting part, I guess, of this presentation is what kind of performance do we see for this system. If we first focus on the analytical performance, we see that there are 2 important papers, one by Professor Christensen and one by Professor Apple. And if you look at the top here, you see that both conclude that the system meets the designation of a high sensitivity troponin assay. And Professor Christensen has focused on stuff like the detection capabilities, linearity, cross-reactivity, etc. Whereas the study from Professor Apple was really focused on determining our 99 percentiles and also the percentage of measurable healthy individuals. And that has been based on the AACC Universal Sample Bank. Zooming in a little bit on one of the IFCC criteria for a high sensitivity troponin assay, you should be able to measure 50% of healthy individuals at least from your LOD onwards. Our plasma LOD is 1.2, and you can see that based on the Universal Sample Bank, we can measure 80% of females and 87% of males. Our 99 percentiles for females was established at 18 nanograms per liter, and for males at 27. And the beauty, I guess, to a certain extent, is that with the AACC Universal Sample Bank, you can make a comparison between different systems. And I think it's good to note that the Siemens systems, all of them, including the point of care solution, meet the requirements of measuring 50%, both for males and for females. So the other criteria from the IFCC is that you should be able to measure your 99 percentile with a 10% CV or less. You can see here that our LOQ at 10% CV is well below the 99 percentile, both in whole blood but also in plasma. So that means that also the second criteria is met. As stressed, we have the possibility to use a capillary, so finger stick sample, which I think is an important feature for a point of care test. And obviously you want to have the confidence that you can use that sample type just as well as a venous whole blood or plasma sample type. So here you see the method comparison, the correlation. You see that the slope and the correlation both are for all sample types are very close to 1. And also if we zoom in on the lower range between whole blood and capillary samples, you see that the correlation holds up very well. So also in that area where your cutoffs are, the correlation is still good. More importantly maybe is the clinical performance. And this has been published by Gonzalez recently. So what they've done is they compared the Teleca VTLI using the 99 percentiles to the Teleca IM lab solution and the Abbott Argitect lab solution. And if you glance over the columns of sensitivity, specificity, PPV, and MPV, you will notice that the values for the 3 systems are actually quite comparable. And that's also why the authors concluded that VTLI demonstrates comparable diagnostics as compared to central lab solutions. So in short, and to summarize, we have published articles that we meet the designation of high sensitivity troponin assay. There's a study done in Saint-Antoine in Paris where they have looked at a method comparison between a Teleca VTLI and a Teleca IM. showing good correlation, and also receiving very positive user feedback from ED physicians and nurses. We have a study where we show the equivalence between the different sample types, and also that there's comparable diagnostic performance if you compare VTLI to the lab. And what's very important to note here is that we have measured in whole blood, and that's also something that some of the guidelines stress, because whole blood is the relevant sample type for a point-of-care solution. So not plasma, but whole blood data is what's required. The future, I think, is quite exciting because the derivation validation setup has been done on a single rule-out for a Teleca VTLI, which I think is very important for this particular solution. And also a derivation validation of a 0-1 hour and a 0-2 hour rapid algorithm as outlined by the EC guidelines will be conducted. So we hope to be publishing on that also very soon. With that, thank you very much for your attention.
So Niels, thank you for highlighting the enormous progress that has been made now with this point of care option, and I was very happy to see that you have the work already lined up in order to do the next step, which of course is necessary to get competitive with the current standard of care in Europe, where using a 0-1 or 0-2 hour algorithm is the standard of care with one of the established assays. So congratulations for these major achievements. Questions, please. Alex, please.
Yeah, really bravo, because this is totally expected since many, many years, and now you achieved it. What was the difficulty to achieve this point of care measurement? Was it because of the whole blood? Was it because you needed to have a small point of care? What was really the challenge?
That's a very good question. So, as I said, I was part of the Philips team, and when I speak to the researchers back in the Philips days, I think they had some difficulties with the magnetic beads and the coating of the antibodies, as well as some of the aspects of the optics. So in the end, those things were optimized, and back in the days when I was still part of Philips, they were already very close to getting a high sensitivity troponin assay, but Philips didn't have the, let's say, the focus or the strategic relevance to prolong the investment. So I'm very happy that they could do that with a private equity behind it, and then being acquired by Siemens.
Richard, please.
No, thank you. I have a practical question. Correct me if I'm wrong, but the point of care would, I think, be very valuable in ED, but my understanding is the 99th percentile, the actual number, is different than the Siemens lab number in the lab. So now in the ED, you're going to be ruling out somebody with a troponin that's high sensitivity, but yet with the same company has two different cut points. Is it ever possible to get the same cut points on the point of care? Because otherwise, I mean, I think it could get very confusing. You know, you have one in the ER and then one measured in the hospital, and that's on a different machine, and it has different numbers. And I just see that there could be some confusion here.
Yeah, absolutely. So if we picture a perfect future, then we would like to align the systems. So a little bit why it is different. We use different antibodies and the technology is different. So we have contemplated on using the same antibodies, but then still the technology would likely lead to differences. And as you know, it's not a standardized assay troponin. That being said, I think there are very interesting projects, for instance, from Professor Mills and Professor Blankenberg, where we look at finding, let's say, risk-based artificial intelligence algorithms, which turn these absolute values into things that you can compare with each other. And I think that's the future. That's where we need to be heading to. But for now, indeed, I think education is very important, and it depends on the preferences of a hospital. So, for instance, you could use point of care only to do the triaging, exclude that low-risk population, which is quite significant, and then maybe follow up the rest with the lab. But yeah, it's definitely not ideal, but I think that's reality for all of us.
Frank, please.
I don't need a microphone. Richard. So I know I'm not supposed to ask questions of the audience, but I'm going to ask this one for you. Richard, do you think that the cut points for point of care and lab-based troponin are the same now? The answer is no. And if that's the case, this is just a better way to skin a cat than we currently do, but no cut point differences. We'll still have differences between the lab and the point of care. Nobody has ever solved that problem. It should not be a reason to slow down point of care.
Yeah, one last question probably is important, the pre-analytical error. We know very well that in the confusion of emergency department, the troponin assay should be influenced by the way that you are taking the blood, and especially with this finger stick, the possibility of hemolysis is there. Do you, because they say that if you send to central lab, they analyze for hemolysis, it affecting troponin, and so do you have this question solved in pre-analytical issue from finger stick that should be influenced the hemolysis?
Yeah, so we have actually looked at the interference of hemolysis, and the beauty of our system is that it is based on the scattering of light. So the magnetic beads will absorb the light source, and therefore it's not dependent on color changes. And that's the theoretical explanation, but also in practice we have seen that hemolyzed samples do not impact our system. So yeah, I think that's we have an answer to that.
Okay, excellent. Thank you very much, Nils. So the old guys now disappear from the podium, and Nisha and Alex take over.
You see that we respected parity, which was not done in the first session. I will let Nisha start. No, I'm sure you are going to do right.
Okay, sorry, just a second. Should I have to press the button?
Yeah, it's already on.
Okay. Can you hear me? That's good. Okay. Hello, everyone. My name is Nisha Renya. I'm from Switzerland, working together with Christian, and it's a pleasure to be here with you all. I would like to introduce the first talk. This is done by Mrs. Danielle Gualando. She is talking about diagnosis and management of PMI. Danielle, please.
And while Daniel is coming, then if we got to try, there are two competitions, one senior lecturer and one young expert.
Sure.
Then Frank is putting all this together. And just as a reminder also for the young experts, later 7 slides, 7 minutes and 3 minutes discussion. I see that Christian tried to influence the chairman.
So, thank you for the opportunity of being here again in person to talk about the diagnosis and management of PMI. So these are my disclosures, but my biggest disclosure is that I'm a big fan not only of soccer, but also of biomarkers, and I really believe that the biomarkers are there to help us to make better diagnosis and treatment of our patients, and between them troponin. And that's my dog, its name is Troponin, and I know that when it gets elevated, it's very dangerous, and mainly if you have food. So
ultra-sensitive troponina, the dog, or all troponina?
Troponina. No, we call it tropo, because you have to be fast, otherwise you don't eat. And the same thing goes from elevated troponin after non-cardiac surgery. So the problem is there's more than 3 million surgeries being performed worldwide, and the mortality related to these surgeries is still higher than expected. The PMI, the perioperative myocardial infarction or injury, is a big contributor to this mortality, and we know it's related to high rates of short-term and long-term mortality. So the goal would be to promptly diagnose a PMI and treat it to reduce this mortality. And for this, I would like to start with an example. There's a very normal case with an old lady, 82 years old, who in the weekend fell and broke her femur. So she came to the hospital. She had already a history of a coronary artery disease with a previous PCI. She was asymptomatic, and she was operated. The preoperative troponin was normal. Then she was referred to the ward. Everything was fine. And the first day after surgery, no troponin was obtained there. And then in the first hours of the second day, she went into cardiac arrest, pulseless electric activity followed by a ventricular fibrillation. She was reanimated, and when the ROSC comes, she had a troponin over 700. Then she was taken to the cath lab. There was occlusion of a marginal artery that was performed a PCI, but unfortunately she died. And when we order a troponin in the blood that was collected the day before, we can see that this value was already by 100. So she had a PMI. So that's the first red flag on the diagnosis of a PMI. There's just no symptoms. More than 80% of patients with a PMI don't have any symptoms, and that is becoming more common the more sensitivity of the troponin assays. And then we search for a second tool as cardiologists, an ECG, and you can stop searching because there's no ischemia signs in this ECG, because only 25% of patients have any kind of ischemia in ECG. Therefore, it's essential to do screening with troponin to find these patients. The second red flag is that up to 50% of these patients being operated will have a baseline level of troponin above the 99 percentile. So we have some kind of chronic myocardial injury. So it's mandatory to have a baseline value in order to differentiate the chronic myocardial injury for the acute injury that happens with surgery. And how exactly to diagnose PMI? So in the Basel PMI study, there were patients included at high cardiac risk submitted to non-cardiac surgery, and the screening was performed with a troponin before surgery and in the first and in the second day after surgery. And if there was a delta here of 14, why 14? Because that's the 99% of this assay, that high sensitivity troponin T, then you would say this patient had a PMI, and he would become automatically a consultation with a cardiologist. Later on, this was validated for another assays, in this case for high sensitivity troponin E from Abbott, with this logic that a PMI requires the doubling of the 99 percentile of the respective assay. And we got exactly the same results. The occurrence of a PMI is related to worse prognosis, short and long term, up to 1 year. But now that we diagnose the PMI, how to treat it? So there's very little evidence about how to treat a PMI. At this point, we're doing some individual approach and multidisciplinary discussion. And why is that? It's because the PMI is a troponin elevation. And in order to treat it, we have to find out the cause and to treat the cause. And what's the cause of a PMI? Here we try to determine and adjudicate why is this troponin elevated. And we can see that that could be, for instance, for extra cardiac causes, and that means not that there's a false positive and the troponin is not coming from the heart, but that the main disease is outside the heart. The classic example would be a sepsis with a secondary myocardial injury, or a pulmonary embolism. embolism, other trauma in thoracic surgeries. And this is only about 13%. Most cases, 85%, we call it, there is a cardiac PMI, and that could be a type 1 or a type 2 MI, or could be other cardiac problems like acute heart failure or arrhythmias, namely atrial fibrillation. That's why there's only retrospective studies that try to describe how they treat the PMI, and there's only 2 studies who try to prove that some strategy works. The first of it, the authors did some propensity match scoring and compared patients that receive what they call intensification of clinical treatment, and that is the optimal treatment with aspirin, beta blockers, statins, and ACE inhibitors, and patients who received that, they had less MACE than patients who did not receive that. The second one is the only randomized trial in this area, the managed trial, in which the authors included patients with myocardial injury after non-cardiac surgery with an ischemic cause and randomized them for the bigatran versus placebo. And in this case, patients with the bigatran, they had less of a combined endpoint for myocardial infarction, but also from venous and arterial vascular issues. So what's the problem is that it's an heterogeneous disease. So to treat it, we have to treat the cause. That's the flow chart that we follow at this point to our patients with a PMI. So first you establish the diagnosis that again is based only in the delta of troponin. No additional criteria is required for the diagnosis. And then we make basic clinical examinations with ECG and hemoglobin, and we look if this patient had, of course, an ST elevation MI or ST depressions with typical chest pain or suggestive of a type 1 MI. We treat them accordingly. If not, we search if there is an extra cardiac cause like sepsis or a pulmonary embolism and treat this cause. If not, we search for acute heart failure or atrial fibrillation. If not, we check the hemoglobin, and if there's severe anemia or bleeding, we correct that. And for all other patients, remains unclear myocardial injury, likely a type 2 myocardial infarction. We give these patients statins, and then if they have no known coronary artery disease, we search for coronary artery disease. If they have non-coronary artery disease, we search for ischemia, and we do an improvement of the clinical treatment. And that's what we do nowadays based on the evidence we have. So, I think it's not working. Can you play the next, please? So we could conclude now that there is two basic commandments of the PMI. The diagnosis is challenging, so I'd say know what you're looking for if you want to find it, and do the screening in patients at risk. And after diagnosis, know what you're treating, because then you're treating it properly. So it's essential to do a good clinical differential diagnosis to find the cause and treat the cause. And with this, I would like to thank all the PMI team and all the people that make possible this research and dedicate their life, part of their lives, or whole their lives sometimes, to help this field that needs help. And at least, I'm not done, please, this last is the most important one, is that a message for Salvatore and the Italian team that the international team has been preparing for years for the soccer match, so they should watch out. Thank you.
We will have many Brazilians in our soccer team this year. Okay, short discussion. Questions? If you can introduce yourself.
I'm Dr. Hyun-Suk Yang from South Korea. Nice to see you. It was a long journey yesterday for me. I arrived here yesterday very, very late.
You didn't come with the metro from Seoul to here?
No, much. The flight. And thank you for the wonderful lecture. And I really agree, perioperative myocardial infarction or PMI diagnosis is challenging. But we should think about the definition of a PMI, perioperative myocardial infarction is also challenging too. So some people just have a definition of increase of troponin, but some people are not completely agree about that. Because myocardial infarction is such a demand and supply impairment or some myocardial perfusion damage. We are always think about myocardial perfusion damage. So if it is not cardiac PMI, I think mostly sepsis. It is not the traditional definition of a myocardial infarction or myocardial perfusion defect. So we should think about the definition of a PMI first, and then we are searching for the diagnostic challenge of PMI. This is my comment.
Thank you. You are absolutely right. The definition of PMI is extremely challenging. What we tried to do is to make something that is easily applicable in clinical practice, because there is other definitions that was also used, like elevated troponin after surgery. But then again, we don't know how it was before. So if you use that, we are including patients with chronic myocardial injury. But I think the main point is to give something that the clinician understands and can use, because if there is a definition, there has 4 lines of definition. A myocardial infarction is something in a patient that don't have a sepsis, that don't have a lung embolism, that doesn't have a trauma, that doesn't have, then nobody will memorize that. So we're trying to create, and we receive a lot of calls because troponin is high. What do I do? Okay, but that's like a severe sepsis. You do nothing. You just treat the sepsis. Yeah, but do I need to give anticoagulants? No, you just need to treat the sepsis. So the idea was to create something easily applicable for the guy that is there at the front.
Thank you. Another question? Okay. Thank you very much. Then we start a journey among the young experts on stage, 7 minutes presentation and 3 minutes discussion. Noemi Glarner from Switzerland will start. Be careful, she's a very good soccer player. Impact of beta blocker on perioperative cardiac complications.
Yeah, thank you very much for the nice introduction. Dear Dr. Renya, dear Professor Mebazaa, dear colleagues and friends, my name is Noemi Glarner, and I'm part of the research group of Christian Mueller from the Cardiovascular Research Institute in Basel. And first of all, I'm very happy that this year we have a whole session on cardiac complications after non-cardiac surgery. And as you heard before, treatment of PMI is still very challenging. So today I want to have a look at the impact of beta blockers on perioperative complications in high-risk patients undergoing non-cardiac surgery. So beta blockers are broadly used in patients with cardiovascular disease because they are known to decrease myocardial oxygen consumption by reducing heart rate and contractile force. So pilot studies have shown conflicting results regarding the use of beta blocker therapy in the perioperative setting. Early trials showing benefits were later retracted due to likely scientific misconduct, and a later large trial using a rather unconventional rapid initiation of beta blocker before surgery showed a reduction in MACE, but an increase in all-cause mortality. So we can say that there are still some controversies remaining regarding the possible benefit of the perioperative use of beta blockers. Therefore, we aimed to evaluate the impact of perioperative beta blocker therapy on the incidence of cardiac PMI within 3 days after surgery and on long-term major adverse cardiac events within 360 5 days in high-risk patients undergoing major non-cardiac surgery. For this purpose, we included consecutive high-risk patients undergoing major non-cardiac surgery at the University Hospital in Basel and at the Cantonal Hospital Aarau, both in Switzerland, and these patients received active surveillance for cardiac complications after their surgery. Furthermore, they were followed up after 1 year. As our primary endpoint, we defined cardiac PMI as a PMI due to type 1 MI, tachyarrhythmia, acute heart failure, or type 2 MI within 3 days after the surgery. And with this endpoint, we can assess very well the potential short-term risk and benefits of perioperative beta-blocker therapy. The secondary endpoint was MACE, and this was defined as a composite of cardiovascular death, spontaneous AMI, acute heart failure, and life-threatening arrhythmia within 365 days after surgery. We used inverse probability of treatment weighting to account for differences in the baseline covariates between patients receiving beta-blocker therapy and those who did not. And in brief, this means that for each subject within the study, there is a weight assigned based on the inverse probability of receiving the treatment that the patient actually received. So this results in a pseudopopulation in which the baseline covariates are independent of treatment status. And we constructed these weights for our model using a multivariable logistic regression model with age, sex, BMI, cardiovascular comorbidities, surgical variables, and preoperative medication as covariates. To assess this model, we compared the baseline covariates in the weighted and the unweighted sample by comparison of standardized mean differences. You will see this again later. After weighting, we performed a univariable logistic regression model with cardiac PMI within 3 days as an endpoint and a univariable Cox hazard model with MACE after 1 year as an endpoint. So in total, we included 7,751 patients, and almost 40% of these patients had a beta-blocker therapy before the surgery. In our cohort, the patients were in mean 74 years old, and 45% of them were female. As you can see here, patients receiving preoperative beta-blocker therapy had more cardiovascular comorbidities like history of coronary artery disease, atrial fibrillation, or hypertension. And as mentioned before, here on the far right, you can see the standardized mean differences for the baseline covariates. And as you can see, these numbers are all very, very high. And while there is no clear consensus on what value of SMD indicates a relevant difference between the baseline covariates, some studies have proposed that an SMD of above 0.1 may denote a meaningful imbalance in the baseline covariates. So now here on this graph, you can see the SMD in the unweighted sample in red, and in the weighted sample in green. And as you can see, with our model, we achieved a good balance in the baseline covariates. So now looking at our endpoints, 812 cardiac PMIs occurred within 3 days after surgery. And when we compare the patients receiving beta-blocker therapy to those who did not, we did not see any difference in the occurrence of cardiac PMI with a weighted odds ratio of 0.99. The composite MACE endpoint after 1 year occurred in about 9% of all patients, and more than 10% of all patients died after 1 year. Here in this graph, you can see the weighted cumulative incidence curve for MACE within 1 year. Again, in red, the patients not receiving beta-blocker therapy, and in green, the patient receiving beta-blocker therapy. And also here, after 1 year, you can see that there is no difference between the two groups, with a weighted hazard ratio of 1.07. The same is true for all-cause mortality within 1 year. Of course, some limitations merit consideration. Unfortunately, we had neither information on the dosage nor on the duration of the beta blocker therapy before the surgery. So to conclude, in our patient cohort, we could not see any difference in cardiac PMI between patients receiving beta blocker therapy versus those who did not, suggesting that continuing beta blocker therapy perioperatively is safe, but was not associated with any benefit. Also, after 1 year, we saw no difference in MACE between patients receiving beta blockers before the surgery versus those who did not. Thank you.
Thank you very much, Noemi. Then you see we could be a very good soccer player and a very good researcher. Very good. Questions? Can I start with one? You followed the patient for 3 days after surgery, is it correct? The heart rate, was it lower in patients with beta blocker compared to those without beta blocker?
That's a very good question, but unfortunately I do not have this information at the moment.
No problem. Yeah, we have a question from...
Sorry, I think I just missed, but I'm Hyun-Suk Yang from Korea. And among your study subject, how many patients got the coronary angiography and how many patients have a positive result in stent implantation?
Sorry, can you repeat?
How many patients among your study subject, the PMI, coronary angiogram, and intervention?
So very little patients of our cohort get a coronary angiography. I think it's below 10% of patients that get this intervention. Sorry?
So we should consider if the patients are sometimes their condition is not good to go to the coronary angiography, I know, but if there is some suspicion of a myocardial infarction, usually we are trying to do at least have some resolve some critical revascularization using the stent. So I'm just wondering how much the percentage of your PMI patients have a angiography. So it is very rare.
Yeah, I think you also need to consider the setting that you are in, because these patients just come from a major surgery, and many of these patients cannot get a dual antiplatelet therapy because of their surgery. So sometimes maybe the cardiologist would advise to get a coronary angiography, but it's not possible. And as I said before, very little patients, I think below 10% or even lower, really get a coronary angiography.
Okay, we need to move on, and just for the young investigators, answering to the question is also very important for the award.
So I would like to go on with the next presenter. It's Hatis Gerhard. She's also from Switzerland, and she will talk about incidence and outcomes of PMI diagnosed by sensitive troponin I. Yes. Please.
Good afternoon, dear Professor Mebazaa, Dr. Arenya, dear audience. My name is Hatice Gerhard, and I'm a research fellow in the group of Basel PMI from Switzerland. And today I would like to present you my thesis with the title Incidence and Outcomes of PMI Diagnosed with Sensitive Cardiac Troponin I. Next slide. I will just talk on. Some background information. So why is this study necessary? Although PMI, thank you. Yes, thank you. Although PMI is known as a perioperative complication and its outcomes were documented several times, there is no consensus definition of PMI. A contributor of this problem is that large cohort studies were giving us guidance for cutoff values using often high sensitive troponin assays. But there are a number of other studies, they are using different cutoff values, different assays. So in this pool of heterogeneous informations, the role of sensitive cardiac troponin I is lacking and made this study for us necessary. Our aim was to look at the incidence of PMI detected by a screening program with sensitive cardiac troponin I and look at the mortality and MACE associated with PMI. Let's look at our patients. Who did we include? We included patients who had a non-cardiac surgery at the Cantonal Hospital Ara, which is in Switzerland, and we included patients with increased mortality risk. So they were older than 65 years or 45 years with a history of coronary artery disease, peripheral artery disease, or stroke undergoing non-cardiac surgery, and they had to stay. They had to have a stay after the operation for at least 24 hours. And we excluded patients with improper screening indications, for example, when they had cardiac surgery within the last 14 days. In our statistical analysis, we wanted to look at the incidence of PMI, and we used the 95% confidence intervals by Agresti and Cole. And to look at the association of the patients with PMI for the outcome mortality and MACE, we did a survival analysis with Kaplan-Meier curves. And in our multivariable regression analysis, we wanted to look how much is PMI as a predictor, how much has it a role on death and MACE as the dependent variables. Our primary endpoint is the incidence of PMI, where the definition with our used assay, the Siemens Dimension Vista, is an absolute increase of 45 nanograms per liters, which is the 99th percentile of this assay. Our prognostic endpoints were MACE, which was a composite of acute myocardial infarction, acute heart failure, life-threatening arrhythmia, and cardiovascular death, and all-cause death. Let's look at the numbers. In the beginning, we had 2,076 patients, and PMI occurred approximately 10%. When we looked at which were cardiac PMI and non-cardiac PMI, there was a clear difference. We had 93% of cardiac PMI versus 7% of non-cardiac PMI. When we looked at the medical record, what these patients told us, only 4% of them said that they had typical chest pain. And I mean, there are other ischemic symptoms, like, for example, dyspnea or atypical symptoms. When we looked at them, they were 15%, and only 2% of them had any ischemic imaging changes. So we were interested how many of our PMI patients did fulfill additional criteria for spontaneous AMI, and there were 26%. But what does this mean? Does it even matter when we look at the outcome death after a year? And you can see in the blue line, this is PMI fulfilling additional criteria, and in green, PMI not fulfilling the criteria. And those two curves are not significantly different. So there was not major variety when we looked at mortality at the end of the year. When we look at all-cause death after a year and compare PMI with no PMI, then the all-cause death was twice as high compared to no PMI. In the group with PMI, there was 90% of the patients with PMI died versus 8.3% in the group of no PMI. This comparison is comparable when we look at MACE after a year. And now let's look to the survival analysis we did. On the left side, you see the cumulative mortality after a year. And here the curves are significantly different. In red, you see the patients with PMI compared to the patients with no PMI in blue. And the same is for MACE on the right side. It's also significantly different. And we did the multivariable regression analysis to look how much is PMI a predictor for those variables. We adjusted them for age, non-elective surgery, revised cardiovascular risk index, and some post-operative complications. Our adjusted hazard ratio for cumulative mortality was around 1.9, and for MACE was it around 3. What were our limitations? As I said in the beginning, we have no universally accepted definition of PMI, which makes it a little bit challenging, and we only had high-risk patients. Screening and treatment, which we did actively, might positively influence mortality and the occurrence of MACE. At the end of my presentation, I want to highlight some of our results. PMI occurred in almost 10%, and only 4% had typical chest pain, so we can imagine that diagnosing them is challenging. All-cause death after 1 year in patients with PMI versus without PMI is twice as high. Our conclusion is, using sensitive cardiac troponin I with this assay, PMI remains an independent predictor of all-cause mortality and MACE. Thank you very much.
Thank you very much. Is there any question? Please.
This very good presentation. Thank you. Thank you very much.
Thank you.
I'm just wondering which is the role of diabetes in predictions of the incidence of, perioperative myocardial infarction, because is it that diabetes is causing no symptoms? So could you please tell us which is the value in your cohort of patients with diabetes? Did you analyze that?
When I look at the baseline characteristics, which I didn't show, then it is, was it clear that it was higher in the PMI group? But this counts also for other cardiovascular risk factors. This was really clear, so we didn't show.
So the influence of diabetes was not more relevant compared to the other risk factor? Diabetes
for
was...
For PMI? Sorry?
No, I mean, in the cohort of patients
yes.
that got myocardial infarction perioperative setting,
yes.
The incidence of diabetes was higher compared to patients that had not? Yes or no?
I didn't check diabetes. Also, in the population of PMI versus no PMI,
yes.
It was higher, yes.
Oh, that's important.
Yes, it was higher.
I think this is something that we should
yes,
definitely...
Yes, when I compared it, yes, it was higher.
And the second question, if I can, could you think that in detecting high-sensitive troponin before making the intervention in these patients at risk with diabetes should be of utility in order to predict and avoid the increased therapeutic approach? Because this is really important to understand which patients can develop increase in high-sensitive troponin. Could you speculate that you can detect high-sensitive troponin in diabetic patients before doing this? Do you think is important, or I don't know, but should be probably something we should work in the future?
It's just a speculation,
yes.
But I think you can speculate that because it, but not always, but sometimes there are like typical patients for that. That's also why we look at the our inclusion criteria in Basel. We later on put the diabetes patients in, but this was an earlier cohort.
Thank you.
Exactly.
Thank you very much. Then it's my pleasure to call Samantha Weder for which patients should receive active surveillance for PMI. Can you do the next?
Forward. Yeah, okay, thank you.
Okay. Dear Professor Mebazaa, dear Dr. Arenya, dear colleagues, I'd like to welcome you all to my presentation about the question which patients undergoing non-cardiac surgery should receive active surveillance for perioperative myocardial injury or infarction. So let's have a look at patient number 1. This photo was taken a few years ago, in 2007 to be accurate. And let's assume during that time that this patient had an accident and has to get a surgery. And let's have a look at patient number 2. It's a slightly older patient who has to get a lower extremity bypass surgery. So who would screen patient number 1 for perioperative myocardial injury or infarction? Please raise your hands. And who would screen patient number 2 for perioperative myocardial? Yeah. Of course, I showed you 2 very illustrative patients. In a daily routine, lots of patients are somewhere in between. And since we have limited resources at a hospital, it's important to find out which patients have at least a moderate likelihood for the development of a PMI. Current guidelines are not very clear whom we should screen, and that's why we wanted to provide precise estimates for the incidences of a PMI for A, specific patient profiles, and B, specific surgical disciplines. So we draw our data from the ongoing prospective and multi-center cohort study, and we included either patients starting at the age of 45 years with a history of coronary artery disease, peripheral arterial disease, or a history of stroke. And we also included patients 65 years or older, irrespective of their medical history. We defined any PMI as our primary endpoint, and for the statistical analysis, we calculated the incidences with their corresponding 95% confidence interval. And in order to justify the resources a hospital uses to screen for a PMI, we assumed that the incidence for a PMI has to be at least 10%. So at the end, we included 9,460 patients, out of which 13.2% developed a PMI. The mean age was approximately 74 years, and 44.2% of the patients were female. So on the left side, you can see all patients with an established atherosclerotic disease, and on the right side, we have patients starting at the age of 65 years or older without an established atherosclerotic disease. We defined this term as either a coronary arterial disease, a stroke, or a peripheral arterial disease, as you can see in the black frames. The red dotted lines show you the threshold we chose of 10%. I'd like to name 3 findings. First of all, you can see that as soon as a patient has an established atherosclerotic disease, the incidences for any risk factor rise. Secondly, chronic heart failure has the biggest impact for the development of a PMI. And thirdly, the more the age increases, the higher the incidence for a PMI gets. Here you can see in the red area we have all incidences for a PMI that are 20% or higher, and the green area shows us all incidences for a PMI that are under 10%, and orange is everything in between. And here I want to point out 2 things. First of all, you can see that patients between 45 to 60 years with an established atherosclerotic disease have an incidence of under 10%. And the same thing you can say about patients between 65 to 70 years without those comorbidities. And as I told you before, we also wanted to look at the PMI incidences for patients by surgical fields. And we could clearly see that thoracic surgeries have the biggest impact to develop a PMI after surgery. And it's also pretty interesting that when we look at the patients without an established atherosclerotic disease, if they undergo a procedure in urology or have a visceral surgery, these incidents are rather low compared to the other surgical fields. I'd like to name two limitations. First of all, we don't have a control group for the patients between 45 to 64 years because we only included patients in this age group with an established atherosclerotic disease. And secondly, the use of the threshold of 10% is of course very arbitrary, but our main aim was just to give a point estimate and then let the hospital or the treating physician decide at what point they want to start a screening or an active surveillance because, as I've said before, and because of the limited resources. So I'd like to draw our conclusions. First of all, we could show that PMI incidences of under 10% are in two age groups. This is in patients between 45 to 60 years with an established atherosclerotic disease, and for patients between 65 to 70 years without those comorbidities. And chronic heart failure seems to have a big impact for the development of a PMI. And also surgical fields can influence the occurrence of a PMI. As I said, thoracic surgery takes the first place. Also we could see that procedures in urology and visceral surgeries, that these specific fields have an incidence of under 10%. So I'd like to thank you all for your attention, and I'm happy to take questions.
Very good, Samantha. Any question? I may start. Oh, go ahead, Veli-Pekka.
Yes, Veli-Pekka Harila from Helsinki. Thank you very much for this informative talk, and I think very clinically important. When you say that chronic heart failure patients has elevated risk, they have the incidence of, let's say, 30% of what you showed. What does that mean? Because they have all the other comorbidities as well. So heart failure patient has coronary artery disease, maybe atrial fibrillation and so on. So how could you say that this is just chronic heart failure?
Of course, you're right. I guess a lot of other risk factors come with chronic heart failure. But as soon as you have a patient in front of you, it's important to note as soon as you know in the medical history that this patient has a chronic heart failure, you know that this patient has a very high risk of developing a PMI. What you're saying, I guess, for the study we did, I guess these numbers are not enough to explain why this is. I guess we should do a regression model to see if what factor has more impact and which one has less. But this study is more descriptive. Yeah.
Samantha, I would like to ask a question on cancer. There is an increasing recent literature showing that patients operated on for cancer have a very high risk of cardiovascular disease, not only related to age, etc. There are probably link between having a cancer and being at risk of cardiovascular disease. Did you see this in your cohort? I think in addition to, because you said thoracic, etc., maybe you should also look on cancer surgery.
Well, we only have data for patients who had surgery because of cancer. So I guess we have a bias in our data, because we don't have any data if the patient was operated with cancer. But when we compared the patients that were operated because of cancer, we didn't see a significant difference. Yeah.
Thank you.
Last question from Detroit.
Oh.
Short question, short answer.
Actually, it's Detroit. I'm just joking. No, thank you very much. My question was, if you know that chronic heart failure is associated with increased incidence of perioperative myocardial infarction, do you think it's because the patients aren't hemodynamically optimized before the surgery? You talk about treatment after. What about treatment before? You know they're at risk, so should they be better evaluated, better treated before they go into surgery rather than identifying them later? Because I assume in a lot of those it may be supply-demand mismatch or something else. And so where they start is important when they get the insult of the surgery on top of it, I think.
Yes, I think you're very right. I mean, I hope that the anesthesiologist who see the patient can figure out how bad if the patient is in acute heart failure. I guess one problem is with urgent and emergency surgeries. There you can't really optimize the patient, but for every elective surgery, I think you're right. Yeah.
Yeah, I'm not sure the anesthesiologist can always do that.
That's true, yeah.
Okay, thank you very much.
Okay, then we move on to the last speaker, and it's really nice to see all these young women on stage. I'm very glad about it. Okay, so this is Silvia Majorano, and she will talk about cardiac complications following emergency surgery.
Thank you very much. Good afternoon, Professor Mebazaa, Dr. Arenya, and their colleagues. Thank you also, Samantha, for introducing my topic of emergency surgeries. So today we're going to take a look at the state of the art. What are we considering nowadays with patients for perioperative monitoring? I took two examples, which are the ACS and skip score and the revised cardiac risk index that, of course, all of you know, to show that some of them, even the more famous, have some limitations. For example, the ACS and skip score, as you all know, is more a surgical predictor rather than cardiovascular. So in our case, it's not so useful. The revised cardiac risk index, which divides patients into 4 classes of risk, is very useful and very used also in the cardiology setting preoperatively. But there are increasing studies that show that it's not sufficient alone to direct the postoperative monitoring of the patients. What's more, our population is a particular population, so emergency surgeries are not really so predictable like the elective ones. So what are our questions? Our questions are, can we imagine that the patients undergoing emergency surgeries unfortunately suffer from more MACEs and from more mortality than elective surgeries? And if so, can we say which patients should be closely monitored after surgery? And are we able to identify a specific surgical discipline or more that have a higher rate of MACE and mortality in emergency setting? So we examined 7 surgical disciplines, which are orthopedics, spinal surgery, thorax surgery, trauma surgery, urology, vascular surgery, and visceral surgery. I'm taking advantage of the former presentations and not presenting the inclusion criteria here, but we used the PMI cohort of patients, in my case from Basel, Arau, and São Paulo in Brazil. 355 were excluded, and then we had 7,500 patients undergoing elective surgeries and 2,850 patients undergoing emergency surgeries. Today we are going to focus on the emergency surgeries which were carried out within 24 hours because we divided the population also into urgent surgeries between 2 and 7 days after indication. Our patients underwent follow-up for 1 year, and our primary endpoints were all-cause mortality, both at 30 days and 1 year, and MACEs, which are a composite of acute heart failure, acute myocardial infarction, arrhythmias, and cardiovascular death, both at 30 days and 1 year. But today we are going to focus on the 30 days outcomes. Here you can see in the baseline characteristics, also like my colleagues before showed, that we can notice in gray overall population, in blue populations who didn't suffer a MACE, and in red those who suffered a MACE. As you can see, the patients who suffered a MACE are a little bit older than their colleagues who didn't, so 75 years in the no MACE population and 78 in the MACE populations. They had coronary heart disease more than the no MACE population, previous myocardial infarction, congestive heart failure, atrial fibrillation, peripheral heart disease, diabetes, COPD, and also chronic kidney failure. And they took more medications regularly for their comorbidities. We then proceeded to examine the incidence of mortality and MACE. This is at 30 days, as said. In red, you will find the MACEs, and in pink, you will find all-cause mortality. As you can see, all of the surgical disciplines are under the 10% of those of incidence, except thoracic surgery, which is a little bit higher in both. So it's 32% in MACEs and 38% in mortality, and vascular surgery, which is slightly higher in mortality at about 12% of the patients. Then we conducted a multivariate Cox regression analysis with which was adjusted for age and sex. As you can see here, for the MACEs at 30 days, thoracic surgery is well above the other curves with an hazard ratio of 22. And the same can be said in the mortality curve, which is all-cause mortality, in which we can also notice vascular surgery, which is significant with an 8.4 hazard ratio. We can say that, so nowadays we don't have a validated tool or score to monitor these patients. The incidence of complication, we can say, in this population was acceptable in almost all of the surgical disciplines. An exception is, of course, thoracic surgery and vascular surgery. What could be a limitation is that, of course, the number of emergency surgeries in the thoracic field were a little smaller in comparison to the other categories. Thank you very much for your attention.
Thank you very much for the great talk. Are there any questions from the audience? Yes, please.
Payman Naziri, I'm from the emergency department, Careggi University Hospital here in Florence. I would like to ask which kind of thoracic surgery are you considering? Is not trauma thoracic surgery, is it included in trauma or in?
It's included in trauma surgery. In thoracic surgery, most of the patients had an emergency surgery due to two different diseases, if we can divide them, pleural emphysema and hemothorax. Hemothorax was due not to trauma, but spontaneous due to pulmonary tumors, or a consequence of a first pulmonary intervention before. So it was a second surgery. But it was divided in this way, so trauma populations are in the trauma surgery.
Thank you.
You're welcome.
Any other questions? Thank you.
Since now we have the coffee break, I propose that we just push by 10 minutes compared to the program. Then I guess we can reconvene at 4:20. 4:20.
To talk. Okay. Okay. Perfect.
Hello, Olga. It's Alex. Yeah, I don't hear you, but we start in 2 minutes. Okay? Thank you for joining us.
Knock, knock. Whoa, that's loud. All right, we've been told that we have to be the time police now since the session before us couldn't seem to know how to manage the watch. So we're going to be good. Dr. Collins, they put two Americans on the stage at the same time. You really screwed up that one. And so now we're let free with microphones. So whenever anybody's going over their talk, Dr. Collins is going to start coughing a lot. You'll think he has COVID. I suggest you get done with your talk, okay? Sean, it's up to you.
Perfect. All right, I believe our first speaker is remote. Dr. Thiel is going to talk about current and future treatment strategies in cardiogenic shock to kick off the cardiogenic shock and AMI session.
in Germany, it's also in Europe. Even last week, I had a 16-hour flight or overall travel time from Dublin to Leipzig. Hopefully, you can hear me. Can you?
Yes, perfect.
All right. Okay. Now I start with my talk on the current and future treatment strategies in cardiogenic shock. So, yeah, these are my disclosures. Mainly the funding we have got for the conductance of the randomized trials we have done and which we are currently doing in patients with cardiogenic shock. And I would like to start with a case which is maybe also helpful for the next speaker, for Veli. This is a patient with anterior STEMI. As you can see it here, he presented, or he had angina since 7:00 p.m. and several hours later he presented to our heart center with these data with blood pressure of 82/55 on a moderate dose of norepinephrine. He was tachycardic. He had elevated lactate. He also had elevated glucose, and he was centralized with cold skin and extremities. And this was a patient from our carpal shock trial. As you can see it here, he had acute occlusion of the LAD, severe stenosis of the left circumflex, and a chronic total occlusion of the RCA. If we have such a patient, we have to make several key considerations. First, is this cardiogenic shock? I think this is relatively clear. Then we have to ask the question, what is the severity? And this will be covered in the next talk. And then we also have to assess, is it predominantly left, right, or biventricular failure? And in the end, we also have to make or answer the question, what are the support options? And this is usually done LV, RV, or biventricular with echo or with a classic Swan-Ganz catheter. When we talk about cardiogenic shock, in particular after acute myocardial infarction, we have a good news that we went down from the previously 80% to nowadays maybe 40% according to registries. And if you look for the large-scale randomized trials, you can see it here, the mortality is somewhere also between 40 and 45%. However, the bad news is that since 20 years, we haven't seen any progress in the reduction of mortality, as you can see it here. It's still in the range between 40 and 45%, even in the most recent published carpal shock trial from our group. Regarding access site, if you take the patient to the cath lab, all the guidelines currently say it's a class 1A recommendation to use the radial access for STEMI, for non-STEMI, as well as for chronic coronary syndromes. And currently, however, cardiogenic shock is only used in less than 20%. This is from our carpal shock trial, where you can see this is adjusted that all-cause death or renal replacement therapy and all-cause death and renal replacement therapy can be reduced if you use radial access in comparison to femoral access. Further supporting that we should use it, however, we have no randomized trials currently in cardiogenic shock for the access side. Revascularization strategy, this is what we have addressed in our carpal shock trial. Roughly 80% of the patients have multivessel coronary artery disease, as you can see it here, from the large-scale randomized trials and registries. And we know from the shock trial that reperfusion, revascularization can reduce mortality. The question is for the 80% with multivessel coronary artery disease, what we should do. Carpal lesion only, carpal plus staged, CABG is also an option, or immediate multivessel PCI. And in the previous guidelines, it was a 2AC recommendation to perform immediate multivessel PCI in these patients with multivessel coronary artery disease in cardiogenic shock. However, evidence or level of evidence C means we do not know it, and that's the reason why we have performed the carpal shock trial, which has been located in all these countries in Europe, and it was also funded by the European Union, as you can see it here. And as you can see it here, there was an absolute 10% reduction in the primary study endpoint, all-cause mortality or renal replacement therapy, from 55 to 45%, which was highly statistically significant for carpal lesion only PCI in comparison to immediate multivessel PCI. And this was mainly driven by an absolute reduction of more than 8%. Number needed to treat of 12 to save one life. I think this is an impressive number, and this is also the reason why in the new guidelines, it is now a class 3 recommendation to not perform immediate multivessel PCI in these patients with cardiogenic shock and multivessel coronary artery disease. So this is a way to go, culprit plus staged. And you should not go for immediate multivessel PCI. There may be some exceptions based on anatomic or angiographic criteria. I already mentioned for the assessment of severity and the cause of cardiogenic shock, we can consider to use the pulmonary artery catheter, the Swan-Ganz catheter, and you all know this, this device or this catheter, which is in place for many, many years or maybe decades. However, data on this are really very limited. Veli will probably show you the data of the Sky Shock 2 definition, as you can see it here. For the assessment of severity, currently, it is recommended to use hemodynamics like cardiac index, pulmonary capillary wedge pressure, based on the Swan-Ganz catheter. You can see it here also for patients in stage D or E. It is recommended to use the Swan-Ganz catheter. However, if you look for randomized data, there's no mortality benefit. The problem with these randomized data, they are not derived from patients with cardiogenic shock. So there were all kind of patients on the ICU, sometimes patients undergoing hip surgery. So we cannot really get evidence from these randomized data. Currently, we have some registry data recently published. You can see it here for patients in more severe Sky shock stage D and E. There is a benefit if we use the Swan-Ganz catheter. And there are also other data, but at least we have data from observational registries showing some benefit. Inotropes and vasopressors, we have this nice table from the American Heart Association scientific statement. You all know this from medical school, all the different receptors, all the different drugs. However, the evidence is really extremely limited on this. We have a subgroup analysis from SOAP2, which suggests that there's a mortality benefit with norepinephrine versus dopamine. However, the overall trial was negative. And we have this trial from France, from Bruno Levy, showing that epinephrine versus norepinephrine leads to same mean arterial blood pressure, same cardiac index. However, epinephrine leads to a higher heart rate, higher lactate or worse lactate clearance, and also to more patients with refractory cardiogenic shock. And that's the reason why this trial was stopped early by the Data Safety Monitoring Board. And if you look into the guidelines, they currently, for vasopressors, give a 2BB recommendation for norepinephrine, and for inotropes, currently, as you can see it here, this is a class C or level of evidence C recommendation because we do not have much randomized data. More recently, published last year in the New England Journal of Medicine, mirikizumab versus dobutamine in patients with cardiogenic shock, single center trial, all kinds of cardiogenic shock, not only patients with cardiogenic shock after an acute coronary syndrome. You can see it here for this, if you ask me, slightly unusual combined clinical endpoint, no difference. And there's also no difference for all-cause deaths, in-hospital deaths between mirikizumab and dobutamine. If you ask me, I go for the cheaper drugs based on this trial. I always go for dobutamine in case I need some inotropes. Last part of my talk will cover mechanical circulatory support. You all know all these devices which are currently available, at least in Europe, some of them, or the majority also in the United States. I do not show the Kaplan-Meier curves of RIBP shock 2 because I've shown it so many times, but I give you the recommendations which are currently in the guidelines. It is a 3B recommendation to not use the intra-aortic balloon pump on a routine basis in patients with acute coronary syndrome and cardiogenic shock. There is a 2BC recommendation for mechanical complications and for patients, for example, as a bridge to destination or bridge to bridge in patient with other causes of cardiogenic shock. Our trial led to an increase in the use of active mechanical circulatory support, as you can see it here in the United States for Impella tandem heart and also for ECMO. I personally believe this is a reflex. Intra-aortic balloon pump does not work, so we try to go for the more advanced and more active devices. Similar data here from Germany, as you can see it here. Intra-aortic balloon pump goes down. We are down to less than 10%. At the same time, increase in ECMO and also a steep increase in the use of Impella, as you can see it from these curves. Now we have to look behind it and looking for the evidence. The problem with that is the evidence is really extremely limited. We had the chance to do an individual patient data meta-analysis. We go back. No difference in all-cause mortality. Some benefits in mean arterial blood pressure, benefits in lactate. On the other hand, as a drawback, we had significantly more bleeding and a strong trend towards more limb ischemia. So some benefits on hemodynamics, on the other hand, more complications. And so probably we have to ask for the sweet spot. When we do not have large-scale randomized data, we usually go for match comparisons, which is the best approach, the next best approach in comparison to a randomized trial. So this is what we did. Impella registry compared match comparison to patient from IVP shock 2. No difference in all-cause mortality with Impella in comparison to control. More complications with Impella, peripheral complications, more life-threatening severe bleeding, and more sepsis with Impella versus control. And then we had in 2019 two large-scale registries, one 28,000 patients, the other 48,000 patients presented at AHA 2019. I show you briefly the results. One registry showed higher mortality with Impella, more bleeding, more acute kidney injury, more stroke with Impella in comparison to intra-aortic balloon pump adjusted analysis, then very highly published. This analysis from Druva, altogether more than 3,300 patients, propensity matched Impella versus intra-aortic balloon pump, once again showing more death, more bleeding with Impella in comparison to control. Now, just a few words on ECMO. ECMO, very limited data, less than 100 patients, as you can see it here. It suggests, based on registry data, that there is a mortality benefit by 33%. In the current guidelines, it has been upgraded that we should use these devices. Honestly, I'm surprised, based on all the evidence, that we could have an upgrade from 2B to 2A, but this is a decision by the heart failure guidelines. Probably most important, if you ask me, this is why we also use it, is patient selection. If you put in such a device in a patient who would survive anyway, you will not change the outcome of the patient, but you probably may lose some patients because of complications. I'm convinced that maybe 20% have a benefit by these devices, but then there are also patients where you can put in the best device on the world and you will not change the outcome because these patients have hypoxic or anoxic brain injury. This is what we call cohort A, no device, cohort B, yes device, and here you can consider to use a device as a bridge to decision. I just, for the sake of time, I just skip this. ECMO venting, this is currently also what is currently discussed. If you use ECMO, you should surely avoid a fully thrombosed ventricle. There seems to be data on observational registries that venting has a benefit for these patients. We also did such an analysis, match pair comparison, ECMO versus ECMO plus Impella. Some call it ECMella strategy, where it suggests, based on this propensity matched analysis, that there is a mortality benefit if we combine it. We currently do a large-scale randomized trial. This is ECA shock, ECMO versus no ECMO in patient with cardiogenic shock. And now coming to the last two slides. Summary, what we currently have as evidence: revascularization can reduce mortality. Culprit lesion only PCI reduces mortality in comparison to immediate multivessel PCI. Norepinephrine, probably better than any other vasopressor. IBP currently or clearly no mortality benefit. And if you are using active mechanical circulatory support in an unselective way, currently we also do not have any hint that this may reduce mortality. And that's the reason why we need these large-scale randomized trials for patients in cardiogenic shock. Currently, in our ECL shock trial, we have a little bit more than 370 patients included. Euroshock, initially initiated from UK, all over Europe, has been stopped. And then we also have the danger trial. Currently, they have 299 patients.
We're going to have to move to your summary slide. I apologize. We're well over time. Okay.
Yeah, this is the last slide. As you can see, these are the numbers, and we will be able to present our results in the next year. And I hope that we will be able to do it at ESC. Thank you very much.
We're gonna have to skip questions and just move on to Veli-Pekka, okay? Talk to us about risk stratification and cardiogenic shock. Veli-Pekka.
Okay, thank you, Mr. Chairman, and thank you, Holger, for starting this important session, although remotely. So just for Holger's case, we do have a risk calculator. It's called cardshock.org, or it's found in the web cardshock.org, and there you can tick all the risk variables and get the number of risk of the patient. So I think Holger uses this every day with all his patients. I hope so. So my only conflict of interest is that I'm really fan of great network, and we started our great study, cardshock study, just because of great network. We showed our protocol 2010, and we've been working very enthusiastically. Regarding cardiogenic shock, the mortality is high, and there's a need for early risk stratification. The patient can be categorized to different risk categories. Already in 1967, Killip and Kimball did their stratification. However, it was a stratification for all AMI patients. So the class 4 was the cardiogenic shock, and the other ones were the milder cases, with class 1 being just clinically compensated patients. A very early risk stratification could aid in clinical decision making, and we really need to know when to increase the intensity of treatment. On the other hand, when are we ready to wean from the intensive therapies, and on the other hand, to avoid futility, so not to start the wrong patients these intensive therapies. The risk scores typically combine several usually independent risk markers, for example, medical history, demographics, the etiology, clinical findings, a lot of routine biochemistry, which we get from our central lab from each of our patients every time they admit, and also scientifically interesting, let's say, novel biomarkers, some of which are coming or are already in our labs, some of which are just waiting for coming clinically available. Then the response of therapy is also one component of some of the scores, which of course you can see by yourself without using any score as such. There are some validated risk scores for AMI, cardiogenic shock, cardshock risk score IABP2, scored by Holger Thiele and collaborators. Also their CLIP score, which I will come back to later on, and a cardshock study included any kind of cardiogenic shock patients. So this risk score applies also to non-AMI patients, which is a difference to the others mentioned before. We already need to have specific risk scores for the contemporary patients using like VA ECMO, and these are examples of the scores dedicated for those patients, but I don't go into details in my presentation. I think this is a good, let's say, white paper of cardiogenic shock. It's a position statement of Heart Failure Association of the ESC, and you can find a lot of important information there. I took this cartoon from that, where you can see a vicious cycle and all different components of cardiogenic shock. Actually, this could be maybe like septic shock or almost any shock, showing these important things like the hypoxemia, acidosis, liver injury, bowel permeability, acute kidney injury, and also thrombotic complications. And in cardiogenic shock, the origin is, of course, the cardiac. This is a busy picture. I'm not quite sure if Holger showed this, and I'm not going to go into details. Many of the green or red boxes are from Holger's ingenious work, where he has done lots of randomized trials in showing us what to avoid and what to do. And you have a lot of these, let's say, these orange or yellow boxes, which we are not very certain. Should we use, they kind of should be considered or may be considered. And I think a risk score helps us in those decision-making processes. Also, one important point is there is the palliative care. Some patients, just to avoid futility, will end up to palliative care. The Society of Cardiovascular Angiography and Intervention classification was mentioned in the previous talk, and I won't go into much details. Actually, it shows a continuum of patients, not exactly yet in the cardiogenic shock, but already, but also at risk, or in those who are beginning, maybe beginning to have shock, because the hypoperfusion is the most important thing in defining cardiogenic or in defining any kind of shock. So here we can see the observed mortality, the intensive care mortality, hospital mortality, and they are very different in these patients, of course. And the extremis patient who has hypoperfusion with deterioration and refractory shock has mortality in hospital mortality of almost 2/3. Here you can see also the, let's say, the distribution of the patients. If you take off my picture for a second, so we can see also the stage E, or we might see stage E, which is just like 1% of all the patients included in this registry. So the most severe cases, happily, are the most rare. This is just to show the variables included in different scores. So we included here in this analysis the sleeper score, which is from the Judith Hoffman's shock trial in the 1990s, the card shock risk score, Holger Thiele's IABP shock 2 score, and their CLIP score, which is most recently published. And you can see that in the left side panel we have, let's say, clinical history or clinical findings, ejection fractions, and so on. And on the right-hand side, you already have biomarkers, quite different biomarkers, and also then findings either in the, I think those days, much by ECG, maybe somewhat also with echoes, and then the TIMI flow, so which is the treatment response. Lactate is the biomarker of hypoperfusion, the most important one. And in cardiogenic shock, in our study with 217 patients having the lactates, the median level was 2.8. And in the quartiles, in the highest quartile, the median was already 8.3, which you saw actually in the SCAI definition that over 8 is typical in the extremis population. You already can see that lactate on baseline is all the time more important prognostic factor than lactate at a certain single time point, even though these lactates also are predictive. But the baseline value is very important and maybe good enough. But then if you think about the relative change, so relative change of course is also a good prognostic factor, so more than 50% decrease. In this figure you can see the Kaplan-Meier curves regarding those quartiles, and in the best population with actually only mildly elevated lactate, the mortality is just like less than 10%, and in those with around 8 you have like 70% mortality. On the other hand, on the right hand side you can see that in those patients who survived 30 days, the lactate goes down quite quickly. There's a huge drop, and these are hours from the start of the study, and in those who died there is much less decrease. These are the components of IABP shock 2 risk scores, and without going to all these details, I just want to highlight that it's very valuable that you classify the patients already by these numbers to low, intermediate and high risk patients, and you can then decide how to use them in clinical practice. Also, you could use them in planning randomized controlled trials, because if you randomize these patients to, for example, ECMO study, you are waiting for much less benefit than maybe in this population in the middle. Are these patients lost anyway, anyhow? That is also a very good question, which I cannot answer. I will now go to CLIP score and just show that also in Holger Thiele's culprit shock study, they analyzed quite a few biomarkers. They had 458 patients, and they analyzed 28 different biomarkers, and out of that regression study they found these 4 markers clinically significant, or the most significant, made a score out of cystatin C, a kidney marker, lactate, interleukin 6, which we heard already in the first session, and NT-proBNP. And here you can also see that you can classify these patients very well. They say that this is a fast and objective biomarker, so you don't need to see the patient at all. You need to just look at the computer, so you can decide which combination you use in your real-life practice. But their message was that biomarkers are very important, and here you can even see that the CLIP score performed almost like better than CLIP score on top of IABP shock score. Antonio Baez Genis did an excellent analysis by making a panel of proteins. They made a mass spectrometry and characterized 2,654 proteins, and out of them, from our CardShock cohort, they found like they looked at 51 protein candidates, and out of them they found 4. And without going to the detail, I think this also shows that sometimes we get results which are not clearly applicable in our everyday practice. So fructose biphosphate aldolase or serbin G1 are not ready in our everyday practice yet. And this is to show that the causes of cardiogenic shock are diverse, so almost or even 40% of the patients can be non-ACS population. So it's important to have a score which includes not only ACS patients but also those without ACS etiologies. We see from card shock risk score that you get 1 point out of the 9 from ACS etiology, but we have a lot important variables. Out of it we can then categorize our patients. Some colleagues are then less enthusiastic maybe and say that whether we are having too many scores but too little information. Of course, like I already before said, we need to do some kind of randomized trials by using the data from these. And sometimes the data from scores is just as good as the clinical gestalt, so your feeling as a clinician. So medicine is not mathematics, but let's see who scores the most on Saturday. Thank you.
Okay, we have time for one question, so make it count.
Yes, Holger's patient got 2 points from card shock risk score.
Thank you. Short question. Lactate is venous, arterial, or is the same? Venous or arterial?
Yes, in most patients it was arterial blood. Actually, I'm not fully aware whether any of the patients had venous blood. So in these intensive care patients, it was drawn from the arterial line.
Okay, thank you. So next we have Alessandra Senzone from Switzerland, but as you're getting ready to talk, Dr. Mueller came up to me and wanted me to remind you that we have an arrangement with the video people, and the clip from that, all the people who present, all the young investigators will get that as to take home with them. I don't know, emailed or something of that way, but everybody gets their own personal clip back.
So, dear Professor Peacock and Professor Collins, dear colleagues, it's my pleasure to welcome you to my presentation about misdiagnosis of acute myocardial infarction, prevalence, patient and institution characteristics, and impact on mortality. As we are all aware, acute myocardial infarction is a major cause of death and disability worldwide. Despite current clinical practice after the introduction of the fourth universal definition of myocardial infarction, where there are multiple investigations available for the diagnosis of acute MI, such as ECG changes, serial measurement of cardiac troponins, and coronary imaging, some cases are still missing. Misdiagnosis of acute MI can have a detrimental impact and harm patients because withholding therapies and procedures such as rhythm monitoring, antiplatelet therapy, high-dose statin, and early revascularization may increase morbidity and mortality. However, to date, there is no sufficient data on the misdiagnosis of non-ST elevation myocardial infarction in the emergency department. Therefore, we aim to determine the prevalence of MIST and STEMI in the emergency department, the potential factors related to misdiagnosis, and to evaluate the impact of possible misdiagnosis on the patient prognosis. So for achieving this, we use the APACE study cohort, that is a prospective multi-center international study recruiting consecutive patients presenting to the emergency department with symptoms suggestive of acute MI. An adjudicated final diagnosis was performed by 2 independent cardiologists, applying the fourth universal definition of myocardial infarction and reviewing all the medical records available, such as patient history, physical examination, results of laboratory testing, ECG, and so on, regarding the patient from the time of presentation to the emergency department until the 90-day follow-up. We use high-sensitivity troponin for our study, in particular, the high-sensitivity cardiotroponin T, the say Alexis by Roche. So from 8,000 to 720 potentially eligible patients, our final cohort was 7,678 patients, as we excluded patients with STEMI and patients with an unclear adjudicated diagnosis and at least one elevated high-sensitivity cardiotroponin T. In our final cohort, 1,363 patients had an adjudicated diagnosis of NSTEMI. So now let's move to the results. So this is the central graph of our study, an alluvial plot that illustrates on the left side the adjudicated diagnosis, so as I said before, performed by 2 independent cardiologists, and on the right side the clinical diagnosis, so the discharge diagnosis made by the hospitals. As you can see, the adjudicated as well as the clinical diagnosis are classified in 5 relevant groups. So NSTEMI in red, unstable angina in orange, cardiac but non-coronary chest pain in yellow, including diagnosis as Takotsubo cardiomyopathy, heart failure, and myocarditis, non-cardiac chest pain in blue, and unknown, unclear symptoms in green. So now following the red flows from the right side to the left side, what we can see and we found out was that 470 clinical and STEMI diagnoses were missed, so around the 35%. So therefore we try to investigate the characteristics of this patient with clinical and STEMI misdiagnosis in order to explore how this patient differed from the others that were correctly identified in emergency department. Of course, I will not go through all of this, but what I can say the most relevant are the age. Our patients were older, 72 years old was the median age. More female, the 34% of our population were women, and with reduced renal function. And what we can say about the cardiac troponin that were used in the different institutions. So as you can see, all of the patients had a measurement of cardiac troponin with the clinically assay available, but as we expected, a large numbers of NSTEMI diagnoses were missed in the hospital that used the conventional one. Then we further classified our patient as either type 1 MI or type 2, and as you can see from our results, the misdiagnoses were around the 50% in the 2 groups. So this brought us to the questions, were these patients undertreated? And here are the results. So these are the following the procedures and the therapies that were carried out during and after the hospitalization. So as you can see, patients with clinical and STEMI misdiagnosis were massively less treated in terms of intervention, only the 38% at the coronary angiography, only the 13% at PCI, and only the 4% at coronary artery bypass graft. And the same about the discharge medication, only the 24% at the dual antiplatelet therapy, and only the 64% at statin. So now to the survival analysis, the Kaplan-Meier curve shows that patients with clinical and STEMI misdiagnosis, so the red curve, had a higher all-cause mortality rate at 2-year follow-up in comparison with the blue one that represent a patient that were correctly identified as an STEMI patient. So this most probably not only because these patients were undertreated, but also because they were sicker. So the alluvial plot showed you that the vast majority was re-adjudicated from an initial diagnosis of either unstable angina or cardiac but non-coronary chest pain. Our study has resolved the study some limitation. Although there was a very detailed patient characterization, there is always the risk of misclassifying a small number of patients regarding the adjudicated diagnosis. And as we excluded patients with terminal kidney failure, we cannot say anything about these groups. So to summarize, the prevalence of missed and STEMI in the emergency department was around 35%. Patients with clinical and STEMI misdiagnosis were mainly elderly, more women with reduced renal function, and particularly in institutions that use the conventional troponin assay. And as I said before, the misdiagnoses were more or less equally distributed in between the 2 groups, type 1 MI and type 2 MI. And also they had a considerable higher all-cause mortality rate. So this brings us to the conclusion that a bundle of measures, including, of course, measurement of high-sensitivity cardiac troponin, is necessary to avoid misdiagnosis of acute MI. I would like to thank all of you for your attention, my entire team of the Cardiovascular Research Institute in Basel, and I will be happy to receive any question.
Great job, and we have plenty of time for questions, actually. Nicely done managing your time. Go ahead. There's a question in the back, actually.
Very nice data.
Yeah. Is that Nick?
So very, very nice data. And a suggestion, really. It's interesting that mice were to type 2s. Clinicians don't like to make a diagnosis of type. And the outcomes in type 2 marker function are much worse. So it would be very interesting to just do a sensitivity analysis by removing the type 2s to see if we're missing type 1 events that we have evidence-based treatment and outcomes could be worse. Have you tried to disentangle that data set yet?
Okay, so not yet, but thank you for your suggestion. I will for sure do it.
If I go to 12, there are much more women misdiagnosed in non-STEMI than men. I think this is very, very important because there is a huge literature showing that there is less women included in trials. And I think this is maybe an explanation. Can you elaborate a little bit more around why there are more women misdiagnosed?
Well, I think that we used to think about myocardial infarction as a male disease, but this is actually not completely true. So, yes, this is a good point. We should maybe include also more women in our studies to have also a big sample and see.
I think all of us, we have also maybe the responsibility to go outside of the cardiological area. Maybe we should put psychologists, sociologists, and many people around the table to try to understand, because here repeatedly we have less ladies diagnosed, less ladies included in trials, and we need really to stop looking only on troponin, etc. There's probably something else that we can and may learn.
Thank you.
Probably one more question. Yeah. Is there, yeah, up front. Go ahead. Yeah, right there. So,
Alessandro, that was a wonderful presentation. Thank you very much. As Alex was saying, that is a very important topic that is coming up repeatedly about women not being represented in clinical trials. Did you look a little bit deeper into the differences between how they presented at the emergency room? Were women repeatedly giving maybe different symptoms? Maybe instead of chest pain, burning, for example. Was that looked at?
Yes, thank you very much for your question. Of course, I didn't have enough time to show you all of our results, but when we also explored the chest pain characteristics, actually you are right. The patient presented with, let's say, atypical chest pain, so the pain was less irradiated to shoulder and neck, and also the main accompanying symptom was dyspnea, so all the characteristics of atypical chest pain. So this is also another point to better go through.
Thank you.
Great. Yeah. Our next talk is on refractory shock, norepinephrine equivalent dose and markers of hypoperfusion by Adrien Picot from France.
Thank you. So my name is Adrien Picot. I'm an anesthesiologist and intensivist in Paris and currently a PhD student in Alexandre Mebazaa's lab. So I guess I'll be the outlier of the day because my talk is actually about refractory septic shock and the prognostic impact of norepinephrine equivalent dose and biomarkers of hypoperfusion. So in critically patients admitted for shock, a significant proportion of deaths are due directly to refractory hypotension or refractory hypoperfusion. And in addition, the lack of response to maximal vasopressor support is a frequent motivation of end-of-life decision, but I'm sure that there could be many definitions of what could be maximal vasopressor support. And this is because the clinically applicable definition of refractory shock is difficult to implement. Theoretically, could be the inability to maintain an adequate tissue perfusion despite some amount of vasopressor inotropes. But the questions are, what are this some amount of vasopressor inotrope, and how to assess tissue perfusion adequacy? And this is probably why most studies dealing with refractory shock have investigated the prognostic impact of of the dose of vasopressor, and have mostly focused on very high dose with the aim of demonstrating futility. But actually, there is a large variability in the outcome associated with a very high dose of norepinephrine, ranging from 60 to almost 100% death. And in addition, it could be interesting to focus on biomarkers of hypoperfusion to have reproducible and quantitative assessments. And of course, lactate reflects hypoperfusion, but also stress and drug-induced glycolysis or mitochondrial dysfunction. And conversely, circulating DPP3 is a new biomarker which has been associated with mortality and increased organ failure in critically patients with shock. And our current understanding of its pathophysiology is that it is released from dying cell. Thus, it could be a candidate biomarker of hypoperfusion. So to explore this question, we used the ADRENOS-1 cohort, which was an observational cohort of sepsis and septic shock patients. There were almost 600 patients with a mean SOFA of 7, and the overall day 28 mortality was 22%. Our primary objective was to explore the relationship between the norepinephrine equivalent dose and day 28 mortality, and to define a cutoff with a prognostic implication, and also to assess the incremental value of lactate or circulating DPP3 on top of the dose of norepinephrine. And importantly, in this cohort, the norepinephrine equivalent dose is the sum of all catecholamine vasopressor used, because there was very few patients with other type of vasopressors. So we first assessed the relationship between the dose of the maximal dose of norepinephrine achieved on day 1 and the risk of death at day 28. And we found that over a certain cutoff of norepinephrine, there was a clear and positive relationship, almost linear, between the dose and the risk of death. But below that cutoff, the relationship was more uncertain. And that cutoff was 0.2 microgram per kilogram per minute norepinephrine, and we used that cutoff for further analysis. And the Kaplan-Meier curves on the right demonstrate the rather good prognosis of patients achieving a low dose of norepinephrine, and the already severely altered prognosis of patients achieving a dose over 0.2. So we next assessed this relationship in the following day, and although the exact shape of the curve slightly differs, there was always the same type of positive relationship. And also the hazard ratio for mortality tended to increase over time, demonstrating that any additional day over such a low cutoff is associated with worse prognosis. And finally, we assessed the added value of biomarkers. And interestingly, both lactate and DPP3 had the same overall behavior, with 3 prognosis groups. First, patients with a low dose of norepinephrine and a low level of biomarker, lactate below 2 millimolar, or circulating DPP3 below 40 nanogram per milliliter. And obviously, patients with both a high dose and a high level of biomarker have the worst prognosis. And interestingly, patients with either a high dose of norepinephrine or a high level of biomarker seems to have the same intermediate prognosis. So in conclusion, over such a low cutoff of 0.2 microgram per kilogram per minute of norepinephrine equivalent, there is a clear linear relationship between mortality for each increment, And we could use that low cutoff as a trigger for the clinician at the bedside to identify at-risk patient, maybe do more investigation aiming at deciphering the mechanism at play, and think about an alternative strategy, let's say another addition of another vasopressor with another mechanism of action, such as vasopressin, angiotensin II. And we could use such criteria for enrichment strategy in clinical trials, testing new hemodynamic strategies. And in addition, lactate of DP3 could be used to further refine the prognosis. And interestingly for DPP3, because it is an enzyme cleaving angiotensin II, it could help define some subpopulations that are more actionable. Let's say to help prescribe angiotensin II, to help define the population of patients that most benefit of angiotensin II, or maybe in the next future to directly target DP3 with an antibody inhibiting its activity. So this is a work in progress, and we are now validating this finding in other cohorts. Thank you.
Great job summarizing a very complex topic in only a few slides. Nice job. Questions from the audience? You talk about enrichment in clinical trials. I think it's a great suggestion. These patients are really, really ill, obviously. What would be the, and you also talk about alternative strategies. So as sort of a trigger of like, you can't just keep ratcheting up the norepinephrine, you have to do something else. What would that something else be if we were to think about even a clinical trial? Would it be another drug? Would it be a mechanical intervention? Like, what are your thoughts?
I think at first it could be used to better define the population who will benefit from another type of vasopressor. There is a lot of debates in the literature in this day about the trigger to initiate vasopressin infusion or angiotensin II infusion, and I think it could be tested sooner in patients with a low dose, already high, but low compared to the existing literature.
Right. Great suggestion. Thoughts, other thoughts or questions? Great, thank you very much.
Next is Paolo Bima from Italy, Switzerland. Diagnosis of MI in patients with malignancy.
So, dear Chairman, dear colleagues, I'm Paolo Bima. I'm actually working at the Cardiovascular Research Institute in Basel. Today, I'm presenting this project, diagnosis of acute myocardial infarction in patients with active or past cancer. So, what do we know? We know that cancer and thrombotic heart disease are deeply interrelated. First of all, because traditional risk factors such as old age, cigarette smoking, obesity, diabetes, they can both cause cancer and thrombotic heart disease. Then we have the fact that cancer activates platelets and promotes inflammation that in turn promote thrombotic heart disease. And then finally, chemotherapy and chest radiotherapy that are used to treat cancer, they can both cause acute and chronic heart and vessel damage. So our working hypothesis was this one, was that what if a patient with active or past cancer presents to the emergency department with chest pain? So based on prior preliminary reports, we would expect higher prevalence of non-cardiac causes of chest pain, such as cancer-related pain. And then we would expect a lower accuracy of high-sensitivity troponin T. So let's jump to the methods. So the cohort that we used was the APACE study that Alessandra has just presented to us. Then the patient, as you said, we enrolled patients with acute chest pain presented to the emergency department, and we excluded patients with end-stage renal disease on chronic dialysis. So we acquired some more cancer-related variables, such as cancer subtypes, date of diagnosis, the fact that it was active, inactive, advanced, or early stage. And then we defined our endpoints. The first one was to define the prevalence of non-STEMI in the cancer population. Then the assessment of the likelihood ratio for acute myocardial infarction of predefined chest pain characteristics and ECG findings, and their dependency on cancer status was assessed via a p-value for interaction. Then we wanted to assess the global diagnostic performance of high sensitivity cardiac troponin T, and this was done via the area under the curve of the RUC curve. And then finally, we assessed the safety and efficacy of the ESC 0-1-hour algorithm. And we externally validated all these endpoints in another international multicenter diagnostic study called the TRAPID AMI study. So let's jump to the baseline. Among more than 8,000 patients, we had 711 patients with active or past cancer. Of those, 592 had a solid cancer, 98 an hematologic cancer, and 13 had both. So about the baseline characteristics, cancer patients were older and had more cardiovascular risk factors, such as previous MI, hypertension, dyslipidemia, and diabetes. The final adjudicated diagnosis, we can see here in brown are always depicted cancer patients. We can see that the non-STEMI prevalence was much higher in cancer patients. And about the non-AMI diagnosis, we saw that cardiac non-coronary causes of chest pain were more prevalent in the cancer population, while non-cardiac causes were less prevalent. Then the predictive ability for AMI of predefined chest pain characteristics and ECG findings, they were all comparable, except for ST depression. As you can see, in cancer patients, the positive likelihood ratio was lower. And here we can see the baseline high sensitivity cardiac troponin T. On the y-axis, we have the levels of high sensitivity cardiac troponin T, and on the x-axis, the final diagnosis. So here we can see that not for non-STEMIs, but for all non-AMI final adjudicated diagnosis, the median troponin value was higher than in non-cancer patients, and sometimes it was higher than the 99th percentile that you can see with these red dashed lines. So consequently, we assess the accuracy of baseline high sensitivity cardiac troponin T via ROC curve. Here we can see in non-cancer patients it was 0.94, in cancer patients it was 0.89, and the difference was statistically significant. Then let's jump to the ESC 0-1-hour algorithm. So for the rule-out group, we assess the sensitivity. And we can see that sensitivity in cancer patient and non-cancer patient were comparable. Then in the observed zone group, we assess the proportion of patient triage to the observed zone. Here we can see that twice as many patients with cancer as without cancer are triaged to the observed zone. About the rule-in, we assess the specificity. So we can see that specificity in cancer patients was lower. All these results were externally validated in a TRAPID AMI study, and the results were comparable to the a PACE cohort. So I want to highlight a couple of major limitations of this study. So causal inference should not be made based on this result. We didn't perform analysis to unravel causality. And unfortunately, no specific sample size calculation was done, so we might be underpowered for certain subgroup analysis. In summary, what we saw was that NSTEMI prevalence was 1.5 times higher in cancer patients than in non-cancer patients. Then chest pain characteristics and ECG findings, excluding ST depression, maintained their diagnostic ability for acute myocardial infarction. Then we saw that the global diagnostic accuracy of high sensitivity troponin T is decreased, as we saw in the ROC curve analysis. And then, most importantly, the ESC 0-1-hour algorithm is very safe to be used in cancer patients. But the drawback is that rule-in specificity is decreased, and also more patients are triaged to the observed zone. Thank you for the attention.
Excellent presentation, and we have a question from one of the great leaders.
Thank you. Interesting data. I think it will be very important if you tell us the difference between chronic cancer and acute cancer, because I think you really put together, you group it two different clinical situations. Do you think do you have this data?
Okay, so thank you for the question. We didn't really define acute and chronic cancer, but we did define actually active and inactive, which is that thing that you say. Yeah. So I haven't presented the data, but actually we did a subgroup analysis in the ROC curve analysis, for example. So for active versus inactive, advanced versus early stage, and we saw that the decrease in diagnostic accuracy was consistent among all subgroups.
So no difference between two groups?
No, we didn't find any subgroups that might have explained that reduction.
Okay.
No, thank you very much. I have a question. Actually, I've brought this up before in the APEX trial. So when you say the people that were entered into it had chest pain, so in the evaluations that we do for high sensitivity troponin, it's always clinical suspicion for AMI, which includes chest pain, but also many other things that clinicians go, it's an old person, it's a diabetic, I don't know. So did all these people have chest pain, or did they have other symptoms? I just would like to clarify that.
Yeah, thank you for the question. So it is true that the main symptoms for which patients were included in the APEX study was chest pain. But it's also true that we also included patients with dyspnea, nausea, and so we have also the data for that.
So I think it would be, in the presentation, we better, the patients that were entered were people the clinician had suspicion for AMI. Because I think when you just say chest pain, it is a little confusing. You know, in our institution, about 10% of patients that come in who have an AMI do not have a primary complaint of chest pain. So
yeah, it's true. Atypical presentations are possible, but then you can, in those patients, you suspect an acute myocardial infarction either with clinical gestalt or after your lab or your thorough clinical and laboratory examinations is done. And this was a prospective study, and yeah, suspicion of MI is true, but it's more true chest pain. So we have also a lot of patients presenting with the atypical chest pain, so left side, right-sided. Thank you.
Okay, we have one more question. This has to be our last one. Danielle?
Thank you for a great presentation. Just one question regarding, have you considered that probably because they go to observe zone, because they may have like chronic elevated troponin levels, maybe because of cancer or because of chemotherapy, and that's why they go to the observe zones? Do you have any data about baseline troponin before they present to the emergency department? Because if they're treated in the hospital, sometimes they do this kind of monitoring. If you do, it would be interesting to compare that.
Yeah, thank you for the question. It's a really good point. In APEX, we didn't record the data about previous troponin levels, but it's true that you can do a kind of similar analysis considering the final adjudicated diagnosis. So it is true that in the observed zone, the most common diagnosis was non-cardiac pain. So maybe they had really a chronic troponin elevation for whatever causes that we don't know from this study.
Great job.
Thank you.
All right, we're going to wrap this session up with Frailty in Patients with Myocardial Infarction by Matthew Lowry from Scotland.
Thank you. Okay, well, hello everyone. My name is Matthew Lowry. I'm a PhD student at the University of Edinburgh. Some of you who were in Rome last year might remember Dr. Christian Nichols talk about frailty and cardiovascular disease, and this is a continuation of that theme. I'm going to present some provisional findings looking at frailty in myocardial infarction. So why am I talking about frailty? Well, firstly, frailty is extremely common, especially in patients with cardiovascular disease. And as our population is aging, it's only going to become more prevalent. It's also a strong independent risk factor of adverse outcomes in multiple conditions, including cardiovascular disease, and is therefore being muted as a way of risk stratifying older patients who present with myocardial infarction. And that group's particularly pertinent because these patients, they're older, they've got more comorbidities, they're complex, they're not recruited to clinical trials, and therefore the benefit and risk of their treatment is not well classified. Oh, excuse me, thank you. But assessing frailty in the acute setting can be very challenging. Traditional frailty measures might rely on lengthy questionnaires, or perhaps getting a patient to a physical test where they walk a set distance against the clock, and that's not appropriate when someone's having an acute myocardial infarction. However, electronic frailty measures, which can be calculated from patients' routinely collected patient data, offer objective and reproducible method for measuring frailty, which could be automated within electronic systems. So we wanted to assess the relationship between an electronic frailty measure and outcomes in patients with myocardial infarction. So to do this, we conducted a retrospective observational study. For a frailty measure, we used the Electronic Frailty Index, which classifies people as frail or non-frail, and if they're classified as frail, then they're stratified into mild frailty, moderate, and severe. And it was validated as a predictor of all-cause mortality in a general population cohort. It's calculated from routinely collected primary healthcare data from roughly 3,000 general practitioner read codes. So we used a data repository containing information of patients in the southeast of Scotland to identify anyone who had a myocardial infarction between 2012 and 2021 using their ICD-10 code. This was linked to primary care data, so we could then calculate their electronic frailty index as well as things like their demographics and comorbidities. We used linked secondary healthcare data to look at how they're managed in hospital, and then national registries to follow them up for 1 year, with our primary outcome being all-cause mortality. So this is the results of the cohort, so I'll just kind of talk you through it. So overall, there were 8,795 patients identified as having a myocardial infarction. If we then compare the frail to the non-frail or normal group, we can see that frail patients are older, they're more likely to be female, they've got a higher incidence of previous ischemic heart disease, and they're more likely to have had an end STEMI than a STEMI. So I think these are all things that we could have predicted prior to seeing these results. But now we'll look at the way that these patients were managed. So on the left-hand side, we have coronary angiography. So you can see that patients who were frail were much less likely to get a coronary angiography, much more likely, less likely to undergo a PCI or revascularization of any kind. And they were much more less likely to be admitted to a cardiology ward. In terms of their outcomes within 1 year, interestingly, they had a non-statistically significant mild increase in terms of myocardial infarction at 1 year between frail and non-frail. But other outcomes were much worse. There's a higher incidence of heart failure, bleeding, and all-cause mortality, all of which were significant. This is a Kaplan-Meier curve showing survival probability at 1 year stratified by the electronic frailty index groups. So you can see it goes normal, mild, moderate, and severe in green, yellow, orange, and red, respectively. And I think there's perhaps two interesting things to say about this curve. So the first one is that you can see for all groups, mortality is quite steep in the first 30 days. After that, patients who have no frailty essentially have very stable levels with a few deaths beyond that, whereas the curves for frailty continue to diverge, and the probability of death at 1 year for patients with severe frailty is about 50%, which is quite stark. So next, we undertook a Cox regression to try and adjust for factors that may be influencing this and to find out what the hazard was that frailty conferred. So this is the univariable analysis, and prior to adjustment we can see that severe frailty carries with it a hazard of almost 10. Next we have the multivariate analysis, which we can see that any degree of frailty still comes with it, confers a significant risk of death, or increased hazard of death, I should say, with severe frailty reaching 3. And as you'd expect, patients that undergo therapy, their risk is slightly reduced, although only coronary artery bypass grafting was significant. So I've kind of rattled through the results, but what are my conclusions? So I think, first of all, frailty is very common in patients with myocardial infarction. 40% of our cohort had some degree of frailty. Secondly, it was associated with a much lower use of what we consider evidence-based treatments like angiography and revascularization. And finally, it was a strong independent predictor of all-cause mortality at 1 year. So I think that this provides some basis to think that frailty could be used as a risk stratification tool for patients with myocardial infarction and help guide treatment in complex older patients. So I'd just like to thank Nick and Atul, my supervisors, and then Chloe and Chris, who helped collate all of the data. And I'd just like to welcome any questions.
Questions from the audience before I let Frank ask one? Danielle.
Thank you for your nice presentation. I'm sorry if I missed it, but how exactly did you determine the frail in an electronic chart? Is that which variables are there? How
Yeah,
is
are we able to bring the slides back up for a sec? So I'll show you, because it can be quite complex. So essentially it uses over 3,000 GP read codes, which are collected through your normal interactions with your GP. It then classifies those into 36 domains, which cover conditions, signs and symptoms, laboratory findings, as well as kind of social factors, whether somebody's admitted to a nursing home or has carers. And then from those domains, it's essentially a cumulative score. So the more domains that you have, the higher your frailty level. And then it just divides them up into the normal, mild, moderate, and severe. So it's something that is calculated from your normal interactions with healthcare, which is why it's useful, for example, calculating automatically when somebody turns up to the emergency department, because it's capturing what they're like prior to their ill. When they're already ill, it's going to be difficult to assess what their level of frailty is.
Oh, Sean's going to let me ask one. I did a study with Oscar Miro a couple of years, same type of stuff, not nearly as sophisticated with electronic frailty index, but in heart failure patients, it's the same thing. If you're frail, you're going to die. But the question is, it's not causal. And the question I have for you is, what happens when a frail person comes in, everybody goes, oh, they're old, they're going to die, and they leave them alone. And I think where we need to go with the data is to demonstrate if they get cath, they live longer or better. And so that's the question I have for you is, do you have anything in your database that allows you to pair an intervention with an outcome in a frail patient?
Yeah, I think that's a really good point. I think one of the things that really caught me there when I was looking at this data is that they had the same rate of myocardial infarction at 1 year between frail and non-frail patients, despite the frail patients having significant less evidence-based treatments. And although I've presented all-cause mortality, what's probably most useful is to look at a cause-specific mortality, so cardiovascular or non-cardiovascular. Because a hypothesis would be that if patients are very frail, frailty is not specific to cardiovascular disease, do they have a very high risk of non-cardiovascular death? And therefore, the benefit that they're going to get from invasive treatment much less. Whereas if they've got a low degree of frailty, there are patients that perhaps would benefit from much more invasive treatment of their cardiological risk factors, as well as undergoing angiography or PCI. So essentially the next stage is look at cause-specific mortality and then look at the factors more deeply, what influences their treatment and how that might affect their outcomes.
All righty. And we are past time, so out of respect for the next session, thanks very much.
So, a quick housekeeping information. The dinner will be ready for you 8 o'clock on the 5th floor. Okay. You take the serious job.
So we have a phenomenal last session for everyone with some really great speakers. So we also have a little bit longer in this session for discussion. So please, lots of questions for our speakers. There is one young expert speaker to come before we go through the senior scientist lectures. And it is from Scotland. And I don't know if you can hear us, but welcome up until 6 a.m. this morning. Anda was going to be here with us in person. So we're all very disappointed you're not able to join us. But great that you're able to give this presentation on COVID-19 and myocardial injury.
That's a good sign. So thank you very much. Many thanks to Valeria and the team for accommodating this. I'm very sad to not be able to join in person, but I hope you are all having a great time and you'll have a great end of the week. So my name is Anja, I'm one of the research fellows. I work with Professor Mill's team at the University of Edinburgh. And today I wanted to present a snapshot of some preliminary results from a study we did in patients presenting to hospital with suspected COVID-19. And we looked at the prevalence and outcomes of myocardial injury. So I don't have any conflict of interest to declare, but grateful to our funders. So just a bit of the background. I'm sure we're all aware that since the start of the pandemic back in 2020, there have been numerous reports evaluating the cardiovascular effects of the COVID-19 infection. Myocardial injury has been widely recognized in patients hospitalized with COVID-19 infection and has been associated with adverse clinical outcomes. At the start of the pandemic in April 2020, we were involved in a global survey which evaluated echocardiogram findings in COVID-19 patients. And our data showed that more than half of patients who underwent a scan on clinical indication had some form of cardiac abnormality. Over time, there has been further data that emerged, including from studies involving cardiac MRI in COVID-19 survivors. The data showed that in patients who suffered a severe infection requiring hospitalization, CMR abnormalities were not uncommon. And there have been multiple mechanisms described, including myocarditis, myocardial infarction, and inducible ischemia. So we were keen to evaluate further these findings and investigate the prevalence, outcome, diagnostic classification, and etiology of myocardial injury in a prospective cohort of consecutive patients admitted to hospital with suspected COVID-19, and to compare the findings in those with confirmed infection and those without, according to PCR testing. So the study was conducted across 3 acute care hospitals in Scotland, and this was done between April 2020 and June 2021. And we enrolled consecutive patients admitted with suspected COVID-19. In addition to the COVID-19 PCR test, patients also underwent a number of hematology and biochemistry laboratory tests done as a part of the COVID-19 order set, and this also included high sensitivity cardiac troponin I. This allowed us to systematically evaluate for the presence of myocardial injury in a cohort of unselected patients. Now, important to mention that the troponin samples were done on surplus blood samples, and the results were not revealed to the clinicians. So we had data from 6,061 patients available for analysis, and myocardial injury, as defined by an elevated cardiac troponin value above the 6 specific 99th centile, was identified in 26% of the overall study population. And you can see on the slide here that the prevalence of myocardial injury was similar in those with COVID-19 compared to those without the infection. So during the initial wave of the pandemic, we systematically evaluated the underlying etiology of myocardial injury in 750 patients with confirmed myocardial injury. We adjudicated the diagnosis according to the 4th universal definition of myocardial infarction using all available clinical data. So you can see here on this panel of sunburst charts that acute myocardial injury was the commonest cause of troponin elevation in more than 3/4 of patients, irrespective of COVID-19 status, whilst myocardial infarction and myocarditis were seldomly identified. Furthermore, the cause of the acute myocardial injury was secondary to an acute illness such as respiratory failure or sepsis rather than a primary cardiac etiology. So we looked at the outcome of all-cause death on follow-up according to COVID-19 status and myocardial injury status over a period of up to 2 years. As you can see from the Kaplan-Meier curves, we encountered high rates of mortality in this population, particularly in patients with myocardial injury and confirmed COVID-19. I wanted to attract your attention to the differences observed here, that in patients with COVID-19, the death outcomes occurred during or immediately after the acute illness period. So we also wanted to look at factors associated with adverse outcomes. And we looked at hospital mortality, in-hospital mortality using an adjusted Cox proportional hazard model. And you can see here the myocardial injury and COVID-19 were independently associated with a 3 times increased risk of in-hospital death. Now, when considered patients who survive the index hospital admission, myocardial injury remains to confer an increased risk of mortality long term. We then were keen to look at subgroups of patients according to their COVID-19 status, so those who tested positive versus those who didn't. And interestingly, we observed an excess risk of in-hospital death associated with myocardial injury again, but irrespective of their COVID-19 status. So in conclusion, myocardial injury is common in hospitalized patients and was identified in 1 in 4 patients hospitalized with suspected COVID-19. The prevalence of similar on those with confirmed COVID-19 and those without the infection. The majority of patients had an acute non-ischemic myocardial injury secondary to their acute illness. The myocardial injury is associated with a 3 times increased risk of in-hospital all-cause death, but this excess risk is not higher in patients with COVID-19 compared to those without. So I'd like to thank the high stakes team and my supervisors, Professor Mills, Professor Newby, Dr. Andrew Chapman, and the data log team for supporting this study. And again, I'm sorry to not be able to be there. Thank you and welcome any questions.
Thank you, Anda. Very clearly presented. Do we have any questions from the audience? I'm going to kick off then, Anda, and oh, here we go.
Thank you so much. I think I came from South Korea. In our country, about 1/3 or more than 1/3 or half of the people in Korean people got a COVID-19 positive, including myself. So for our country, I think COVID-19 positive or negative evaluation is very hard because most of the people already have a positive. So I think in this study, they said 1/7 people have a severe LV dysfunction. I was so shocked. So I think for analysis, the severity or comorbidity of the people who have a COVID-19 positive is much more important for the impact of myocarditis or other things, I think, because it's so common in Korea. About half of the people is a COVID-19, so COVID-19 is not equal to the myocardial injury or myocarditis. I think comorbidity is much more important. So just to let me know what is the comorbidity of this kind of cohort. Thank you.
Thank you very much for this very good question. So yes, I think I agree with your observation. So in this study, we included only those patients who were admitted to hospital with suspected COVID-19 infection as one of the surrogates of the severity of the infection. So we didn't include anyone in the community. I don't have yet data to look at the severity of infection in terms of admissions to ITU, but something that we'll look into in the future. In terms of the comorbidity question, so we looked at the 4 subgroups, those with myocardial injury and COVID-19 and those without. And we did identify that those with evidence of myocardial injury and COVID-19 were older patients, and those had higher degree of underlying comorbidities, particularly cardiovascular comorbidities, including ischemic heart disease and cardiovascular risk factors such as diabetes, hypertension.
Even that COVID positive or COVID negative had similar long-term prognosis, but it seemed that COVID positive had a very high mortality at the beginning, while the COVID negative were more progressive. I think it's different, the course of these 2 situations.
Exactly, yes. So that was, hence we decided to look at this by stratifying them according to outcomes during the index hospital admission, and then in those who survived the index hospital admission. And I think it's very interesting also that the hazard ratio in the survivors for COVID actually looks to be protective for outcomes, which tells us that those patients who survived the episode, they actually have a good baseline and less risk factors for death overall.
Okay, thank you very much for joining us, and I really appreciate an excellent presentation.
Thank you.
Already warming up in the back. The next speaker is Andrew Chapman. We are now going to the senior scientists lectures. All of these gentlemen and one lady are either very senior in their research or have won one of the great research awards before. So there's every reason you can be excited to hear the latest research. So Andrew, you talk about the challenges in type 2 myocardial infarction. So what a nice continuation after the previous talk. Andrew.
Thank you, Professor Mueller. Ladies and gentlemen, it gives me real pleasure to be here in Florence today, the heart of Europe, on the day that Boris Johnson got fired. And as you've heard, I'm Andrew Chapman. I'm a clinical lecturer in cardiology from the University of Edinburgh. And I have a lovely topic to discuss in just 10 minutes. What are the challenges in type 2 myocardial infarction? So it's difficult to know where to start, but I'll try and give you an overview. We all are familiar with type 1 myocardial infarction, an event which occurs due to an atherosclerotic plaque rupture, intraluminal thrombosis, distal ischemia, and infarction. But the universal definition has introduced a concept of type 2 myocardial infarction, which we can think of as an umbrella term which encompasses coronary mechanisms of MI, which can include spontaneous coronary dissection, embolism, vasospasm, and it also includes patients with fixed obstructive coronary disease and patients who may have no coronary disease at all. So it really is an umbrella term. And we know that the prevalence of type 2 myocardial infarction varies depending on whether we check troponin levels in all patients who come into hospital or if we only check troponin levels in patients who are very unwell with another problem. And as illustrated on the right-hand plot here, there are a number of different mechanisms of type 2 myocardial infarction, and the systemic mechanisms may include hypotension, prolonged tachycardia or tachyarrhythmia, or indeed hypoxia. Outcomes for patients with this condition are very poor. We've previously demonstrated that patients in blue with type 2 myocardial infarction, as few as 1 in 3 are actually still alive at 5 years after diagnosis. And when we look at the specific cause of death, we can see that the future event rates of type 1 myocardial infarction or death from a cardiovascular cause are still quite high. So that's shown in gold here on this panel, with patients in red having a previous type 1 myocardial infarction. And that is despite this significant excess risk of non-cardiovascular death, similar to the response Dr. Lowry gave you regarding frailty. When we look at the event rates of future type 2 myocardial infarction in patients who have already had a type 2, they are at 6-fold increased risk of having a recurrent event within 1 year. So these are patients with real cardiovascular risk. And what is the challenge? Well, let me illustrate that for you with a diagnostic case. So this is a middle-aged male patient who presents to hospital with fever, breathlessness and cough, having been breathless for a couple of days. And he describes an ischemic sounding chest pain. This is his ECG, which demonstrates, I hope you'll agree, a left axis deviation, a left bundle branch block, and a tachycardia in sinus rhythm. He's febrile at 38.9, he's got an oxygen requirement, he has a CRP of 160, he's got a troponin of almost 6,500, and he has evidence of consolidation on a chest x-ray. So if I can just ask you for a show of hands, do you think this patient has a type 1 myocardial infarction? A type 2 myocardial infarction? Anyone? Acute myocardial injury? Okay. So he had chest pain. So he's had ischemic sounding chest pain. He's got a troponin rise of 6,500. And he's got an ECG that demonstrates a left bundle branch block. So yes, he does have evidence of ischemia and a troponin rise. Okay, so there's some voting for type 1, most voting for type 2. So this patient was enrolled in a study called DEMAND-MI, which was the first prospective observational cohort study in type 2 MI, and he had an invasive coronary angiogram. The left panel demonstrates his right coronary artery, which is subtotally occluded. The central panel demonstrates that he's got a severe lesion in the proximal left anterior descending, and the right panel demonstrates the LAD in a vertical plane, and again he's got this very tight lesion at the top, just coming in here. And this lesion was interrogated using a technique called optical coherence tomography. This gives you a bird's eye view down the coronary artery, and it allows you to look at plaque morphology and evidence of clot. And it should keep playing, and hopefully what you'll appreciate just towards the bottom is that there's a plaque rupture and there's some clot and thrombosis here. And on the right-hand panel, he's had a cardiac MRI, and he's got late gadolinium enhancement in an area consistent with the LAD territory. So this is a patient that we now know has had a type 1 myocardial infarction, and it's important to remember that you have a pneumonia, you have a pro-inflammatory condition, and your risk of a plaque rupture MI is actually 3-fold higher within the first 90 days. And this is one of the challenges of type 2 myocardial infarction. So in DEMAND-MI, we took 100 patients with a clinical diagnosis of type 2 MI, and we really carefully selected these patients. And we got it wrong. We got it wrong 7 times. So 5 patients had plaque rupture or thrombosis, 1 patient had myocarditis, and 1 patient had a stress cardiomyopathy. So these are very difficult distinctions to make in clinical practice. And the guidelines talk about these conditions as 2 separate entities, but in actual fact there is undoubtedly a grey zone. And the use of different imaging modalities can certainly help us to try and integrate and differentiate between these conditions. But at present we don't really have an optimal approach as to how to do this best. So which patient should have a coronary angiogram? So the guidelines say that if there's diagnostic uncertainty and ambiguity about whether someone might have had a plaque rupture or thrombosis, then they should have an invasive coronary angiogram. We know in studies of consecutive patients that this very rarely happens in practice in type 2 MI. Less than 10% of patients will come forward for angiography. So patients who have a higher peak troponin, patients with limited supplier-demand imbalance, patients with persistent symptoms or very clear regional ECG changes might be more likely to have an acute coronary lesion, and perhaps they're the patients you should be considering taking forward or having a lower threshold. Those who are older or more frail or who have more generalized patterns of ischemia, or if it's short-lived in the context of a very significant stressor, you might be less inclined to take them forward. But that's a non-evidence-based recommendation. Can novel imaging techniques help? Well, this is 2 different techniques which are in development at the moment. On the left side, there is an 18F GP1 thrombus tracer, which is essentially looking for evidence of clot. And the lower panels demonstrate here clot in the circumflex artery and also clot in the left atrial appendage in a patient that presented with rapid atrial fibrillation. So he's had a presumed embolic event. So this is a research technique which may well be interesting in the future. And we also can use CT imaging to look for markers of plaque characteristics. So we know low attenuation plaque is highly prevalent in patients with type 1 myocardial infarction and less prevalent in patients with type 2 myocardial infarction. And that might also be a helpful non-invasive adjunct that we can use. Can biomarkers help? This is predominantly a biomarker meeting. Well, researchers from the APACE group have previously shown us that some of these biomarkers can discriminate between type 1 and type 2 events. These are predominantly markers of vascular stress or atrial stretch, rather than any specific marker related to the pathology of type 2 myocardial infarction. And it's also been shown by other groups that these biomarkers can help with discrimination, predominantly in the research area at the moment, though. And thinking about susceptibility to myocardial infarction, well, there are patient factors related to the illness. There are undoubtedly coronary factors related to whether someone has undiagnosed coronary artery disease. And there may be structural factors if patients have got undiagnosed hypertrophic cardiomyopathy or valvular heart disease. And I like to think of coronary disease as a bit of a spectrum. So hopefully myself, I don't have any obstructive coronary artery disease. So if you give me SVT and my heart rate goes 190, I hope I could tolerate it for a reasonable length of time. If you turn that on its head and you take someone who's in their 70s who may have undiagnosed obstructive coronary disease, if they have atrial fibrillation for a shorter period of time, they might develop ischemia. So it's thinking about the underlying ischemic substrate and how we might modify that in future risk, I think, is quite important. And in DEMAND-MI, again, we looked for the prevalence of underlying cardiac disease. So 1 in 3 patients with type 2 myocardial infarction in our study had obstructive coronary disease. And 1 in 3 patients had evidence of LV systolic dysfunction. And these were previously unrecognized in over half of participants. So a very high burden of undiagnosed cardiac disease. And we want to try and understand if we can deliver a randomized trial in this population. So I've been funded in Scotland, and I'm very pleased to say that we will be able to recruit 60 patients with type 2 MI, and we're going to randomize them 1-to-1 standard care or intervention, which will be a consideration of cardiologist assessment, determination of likelihood of LV impairment or coronary disease, and appropriate investigation and secondary prevention. And I really hope that this will be a step towards a large international multi-center trial that we can power for clinical outcomes. So in the last 20 seconds or so, just to conclude, type 2 MI is highly prevalent in clinical practice, but we know this is a real problem. We don't have sufficient prospective evidence in order to tell us what to do with these patients, but we're trying. We think imaging with CT coronary angiography, invasive angiography where required, and plaque imaging can be crucial in clarifying the diagnosis. There's undoubtedly a large burden of unrecognized coronary and cardiac disease, and randomized controlled trials are the way forward that will allow us to demonstrate benefit, hopefully, and direct our future efforts. Thank you very much for your attention. And if I could just put that last slide up, just as a first time in the senior investigator session, I really do have to acknowledge the hard work of the fellows that were on my slide, particularly Dr. Vilarga, who can't be here today, Dr. John Hung, and many others you'll see. Thanks again.
Fantastic, thank you. Questions on type 2 myocardial infarction? Until the first one from the floor, I may start. The universal definition of MI would argue that the first patient that you have presented because he had fever and systemic infection might not be considered type 2 myocardial infarction because this might be septic or whatever. So how strict are you with your definition? Would a patient with fever, if he's tachycardic, still qualify for type 2, or should he be called kind of cardiac other injury? How do you use the term in clinical practice?
Excellent question. So I think it comes back to where is the evidence for supplier-demand imbalance. So that's a patient who's got clear evidence of hypoxia and tachycardia and a febrile illness. So you could potentially argue it in either way. I think at the moment there's no consensus as to the use of fixed criteria. Professor Tegeson and Dr. Sabi, that he used to work with, did previously propose very strict criteria for supplier-demand imbalance, but we don't use them in practice for the reasons I illustrated around the case with SVT and atrial fibrillation. So I think having met that patient in practice, he clearly described ischemic chest pain radiating to his left arm, his neck, his jaw. in the context of supply-demand imbalance. So I was quite comfortable that the clinical diagnosis was type 2 MI, and his imaging evidence supported that rather than myocarditis.
Payment, please.
Thank you for this presentation. My question is related with emergency department always. In patient with acute atrial fibrillation, do you suggest to measure troponin to every patient, or do you suggest to measure it in some patients only?
So it's a difficult question, it's a challenge. I think if a patient has atrial fibrillation and clear evidence of sustained ischemic symptoms in the context of a lower rate than you might anticipate to cause those symptoms, or in someone who's got clear regionality to the ischemia on the ECG, I think it's reasonable to look to see if there's evidence of myocardial infarction. I think measuring troponin concentrations in all patients with atrial fibrillation, you'll undoubtedly pick up some patients who perhaps have subclinical ischemia and they have a small elevation of cardiac troponin and you might not be sure what to do with them. So my suggestion as a pragmatic level would be if someone's got sustained ischemia at a low rate or very regional ischemia, then consider troponin.
Thank you very much, Andrew. Excellent talk, as always. So in my clinical experience as a cardiologist registrar, it's sometimes very frustrating because you have to see many patients in the emergency department which were diagnosed with type 2 myocardial infarction, although they do not have clear signs of ischemia. So I found it personally very, very difficult to detect objective ischemia. So what's your personal opinion? What should be the next step? Which patients should really get an echo, for example, to look for regional wall motion abnormalities? What's your opinion on that?
Thanks, Jasper. I think the ischemic criteria are very pragmatic, and I can see why they were attractive when the guideline was drawn up, because we understand ischemia, we have criteria for myocardial ischemia in our head, and we can see those patients. But actually, when you look at the totality of the evidence, comparing patients with type 2 myocardial infarction and those with acute myocardial injury without ischemia, the rates of future myocardial infarction, cardiovascular death, are higher in those who have acute myocardial ischemia. And that might be because at the moment we're ignoring those patients and we're not giving them the same level of investigation and treatment. So that's my hypothesis, and certainly going forward, one of the studies I'd like to do is looking at the role of ischemia in the diagnosis of type 2 MI and acute myocardial injury. Does it actually differentiate the presence of obstructive disease, late gadolinium enhancement, or indeed inducible ischemia, or perhaps could the classification be refined?
So thank you very much. It's a very complex, confusing area. So what we've been looking at is biomarker combination, but the theory is that if you develop supply-demand mismatch for whatever reason, that will occur over some time and you get wall stress, and then that wall stress will eventually result in injury or whatever you want to call it. So we've been looking at the NT-proBNP to troponin T ratio. Mm-hmm. And what it appears is the higher that ratio, the more likely is that it's supply-demand mismatch. And while it's not perfect, I think it's a sort of an interesting way to try to differentiate, because the mechanism is different. And the type 2s don't come in and say, 2 hours ago I got sudden onset, I didn't feel well, I got pneumonia. And so I think that maybe biomarker combinations maybe, and maybe you would like to look at that in your...
Certainly, it's an interesting area, and I think what you're describing is the correlation between whether someone has stress and has another insult leading to that stress, which can be associated with the type 2 MI. And one of the great things about Demand MI is that we were funded to bank samples, so we're very interested in looking at these things in future.
Okay, well, if there are no more questions, then we'll move on to the next speaker. But Andrew, thank you very much for that excellent talk. So our next speaker will be known to many of you already. Jasper is joining us to talk about the Precise MI trial. There is not an error in the program. You may have noticed that Jasper here is from Scotland. And after attending many great meetings, we decided on who we were going to transfer and who we weren't, and we chose Jasper. He's now speaking to you as a card-carrying Scottish cardiologist. Welcome, Jasper. Looking forward to your talk.
Thank you very much, Nick. Thank you very much, Christian. Dear friends, it's a great pleasure to be back to the stage here at the great meeting, to see so many friends and known faces. So as Nick already highlighted, for me it's a very special great meeting this time because there was a huge change in my life actually. So my family and I moved to Edinburgh this year. And so why did this happen actually? So I'm very happy to explain. So this is me, obviously. I'm a German potato, so that's how we call ourselves, a German potato. So I grew up in northern Germany, in Hamburg, so quite close to the sea. But I decided to move to Switzerland to join Christian's group, and it was a very pleasant time because, as you may know, Swiss people, they love potatoes, okay? So in every way. Not specifically German potatoes, I have to say, but potatoes, especially in combination with cheese. But with the time, after a few years, I really started to miss the sea because I grew up in Hamburg, so close to the sea. So I thought, where can I continue world-class research with people who love both potatoes and fish? And that's the solution. It's Scotland. It's fish and chips. So this is the place to be for me, close to the sea in Edinburgh, so I love it. Anyway, so for today's presentation, oh, that's the new me, by the way. For today's presentation, we are going back to the Swiss Alps because, of course, I haven't left Christian. I would never do. So we continue our research together, and I'm happy to present the PRECISE-MI trial. So PRECISE-MI stands for the Prospective Evaluation of the European Society of Cardiology's 1-hour algorithm safety and efficacy for the triage of patients with suspected myocardial infarction. And this is a prospective step-batch cluster randomized controlled trial. So these are my disclosures. So the triage of patients presenting with a suspected myocardial infarction to the ED is still a big challenge for all of us because we have about 20 million patients presenting to the ED in North America and Europe every year. So in these patients, we need an early triage decision, and we know that only a minority of chest pain patients finally are diagnosed with acute myocardial infarction. And we are able, by using current clinical pathways, to rule out an acute myocardial infarction in the majority of patients. The guideline recommendations have changed over the last decade. So since 2011, the ESC guidelines recommend to use the ESC 03-hour algorithm, so to perform the second blood draw 3 hours after ED presentation. This is the ESC 03-hour algorithm, the concept from the guidelines. You can see on the left side, indicated by the green arrows, the rule-out pathway. On the right side, indicated by the red arrows, the rule-in pathway. And the important point here is that the ESC 03-hour algorithm uses assay-specific 99 centile that were derived by the diagnostic companies in a healthy reference population. And on top of that, if we want to rule out a patient with the ESC 03-hour algorithm, we have to use a formal risk score, which is the gray score, with a score of less than 140. So guidelines changed, and since 2015, the European guidelines now recommend the ESC 01-hour algorithm. And as the name indicates, we should perform the second blood draw 1 hour after ED presentation. And these recommendations are still the current recommendations in the last update in 2020. So this is the concept of the ESC 1-hour algorithm, just as a quick reminder. So what we have to use is, first of all, the ECG to rule out a STEMI, of course. And then we have to apply the 1-hour algorithm to non-STEMI patients or patients with a suspected non-STEMI. So we use very, very low baseline concentrations for direct rule out of patients if the chest pain onset is more than 3 hours ago, or alternatively, we use low delta changes between the 1-hour and 0-hour concentrations. On the other hand, we have very high baseline concentrations for direct rule in or a significant change, and then there's a third observed group of patients who then need, for example, resampling of cardiac troponin. So important to highlight is that the cutoffs to use are assay-specific again, and they're available for most high-sensitivity assays that are on the market. This is the central table from the guidelines giving us all the specific cutoffs that we should use for rule out and rule in of a non-STEMI. However, there are major gaps in knowledge, and that's why we set up the PRECISE-MI trial, because the evidence that we have today, mainly based on prospective cohort studies with central adjudication, and there's actually only one randomized trial from 4 Australian sites. So we actually don't know if it is feasible to implement the ESC 01-hour algorithm in very busy EDs worldwide across different healthcare systems. We also don't actually have data from a randomized trial on the direct comparison of guideline-recommended pathways. As I highlighted before, we have different high sensitivity assays and different EEC 1-hour algorithms, so there's no trial comparing these different 1-hour algorithms with each other. So finally, there's no international multi-center trial across different countries and healthcare systems. So that's why we decided to start the PRECISE-MI trial. We started recruitment in December 2020, and the primary aim of the study is to directly compare the safety and efficacy of the 1-hour algorithm with the 3-hour algorithm. The design, as I mentioned before, is this prospective stepwise cluster randomized controlled trial, and we've chosen this design wisely in accordance to the trials that were performed by Nick Mills and his team in Edinburgh, so the historic and the high-stakes trials. The inclusion and exclusion criteria are very simple. So we include centers that are currently using this 3-hour algorithm and that are willing to implement the 1-hour algorithm in the near future. And we enroll adult patients that present to the emergency department with non-traumatic acute chest pain or discomfort and a suspected acute myocardial infarction. As done in previous studies in this area, we exclude patients with kidney failure requiring dialysis, patients presenting in cardiac arrest or shock. There are two co-primary endpoints, actually. We have a safety endpoint on the left-hand side, which is all-cause mortality or type 1 myocardial infarction within 30 days. On the other hand, we have an efficacy endpoint, which is the time from ED presentation to either discharge home or transfer within the hospital. We anticipated a MACE rate of 3%. We use a non-inferior margin with an odds ratio of 1.3, and therefore we have to enroll 52,000 patients to show non-inferiority regarding safety and superiority regarding efficacy with a power of at least 90%. There are several secondary outcomes. I will not talk about them in detail, but as I highlighted before, it's also about the feasibility. So is it really feasible to perform the 1-hour concentration within a specific time window, around 1 hour after ED presentation, for example? Then there are, of course, also, we will investigate the MACE rates at 1 year, for example. So we already set up an international consortium. We have 20 sites at the moment from 10 different countries. Not all of them are active at the moment, and they start recruitment in groups, as I will show you in a second. So this is the stepwise design that we are using. So in December 2020, 6 centers started enrollment. And first of all centers continue to use current practice, which is the EEC-03 algorithm. After 6 months, we then group the participating centers of this group according to the expected number of patients enrolled. And then we randomize which of this group starts early versus late implementation. So the first group then run a blanking phase, which is something like a transition period. And during this blanking phase, we perform standardized teaching with posters, for example. We train the local PIs who will then continue teaching their local staff. Afterwards, the early implementation group starts to implement the 1-hour algorithm, while the other group continues to use the EEC-03 algorithm, then perform the blanking phase, and then, as in the late implementation phase, implement the EEC-01 algorithm. In all patients, we perform 2 different follow-ups, a 30-day follow-up and a 1-year follow-up. It's important to highlight that we were able to get ethics approval for a 30-day follow-up without patient-level informed consent at most centers, and for the 1-year follow-up, we have to seek informed consent. There are some methodological strengths that I would like to highlight. So first of all, we can use most data, or we only use administrative data, and we were able to set up collaborations with local IT teams to automatically extract most of the data, then transform the data and implement them, upload them automatically into our database. It's a very pragmatic and efficient design, actually, that we've chosen. And we have a great team in Basel, more than 10 people now, who perform automatic range checks all the time, logical data cleaning. So we're really targeting to have the highest data quality possible. The MACE events rate will be adjudicated by independent adjudicators with a blinded to study phases, and with a data safety monitoring board, which is led by Professor Mills. So there is a huge relevance for research because we will, for the first time, generate evidence in this field of research from a randomized trial. We hopefully can reduce overcrowding in the ED, which is a worldwide problem. And finally, we hope to reduce healthcare costs. So this is the international consortium. I would like to thank all the PIs for doing just an exceptional job. We're able to enroll 22,000 patients already. So there's a way to go still. And the third group of centers will start by the end of this year. And we're still open to include new centers and actually very keen to welcome new collaborators. So we want you for the Precise MI study. The nearest recruiting station is here or here, so here, there. So please feel free to contact us. And finally, I would like to thank the outstanding team which is behind the study, the Precise MI team in Basel. Thank you very much for your attention.
Thank you, Jasper. Really important trial and phenomenal job in getting it set up and recruiting well. I have some questions, but Luella, do you want to kick off?
Thank you. Really nice study, and it's nice that my site is able to participate in it. I've got a question for you about efficacy, because of course exit block is an international problem, and it's only gotten substantial. So that's the discharge or transfer from ED, and of course this is only worse post-COVID. And certainly in my hospital, which is relatively efficient, we now frequently have patients in the emergency department for 36 hours plus. So obviously that's going to play havoc with that endpoint. What are you doing to mitigate that in the follow-up?
Yeah, that's a very good point. So we were discussing a lot what to do with COVID. So what we're definitely doing is to ask centers to provide like key numbers for COVID, like monthly performed tests, hospitalized patients, so to try to adjust for this. Otherwise, it's a very important point, because as healthcare systems are different, there are emergency departments as yours, where patients are kept for quite a long time. So we actually set like a limit of 12 hours here, and then have to see whether we perform a sensitivity analysis in these patients.
Super. I have a question for you, just as we're waiting for others. I was just pondering it while listening to your presentation. Your safety outcome of MACE is interesting, because your pathway isn't just an early rule-out pathway. It provides guidance for high-risk patients who are ruled in, which is not the case with the standard 0-3-hour pathway. So is it feasible that the implementation of the pathway will not just be safe, but actually more effective and reduce MACE? And how are you going to address that from an analytical perspective if it were to happen?
Definitely. Thanks, Nick. So this is one of the secondary outcomes that we will investigate to see whether we can also increase, let's call it the accuracy for rule-in, and if we will treat more patients. What's the time, for example, from presentation to transfer to the cath lab? So we definitely investigate this. But you're absolutely right. It's really important to highlight that the 1-hour algorithm is not only a rule-out algorithm, so it might change how patients not only ruled out but also ruled in. And just as a reminder, the 3-hour algorithm uses the 99th centile, so it really detects myocardial injury, while the 1-hour algorithm uses a much higher cutoff and will maybe really identify more high-risk patients there.
While I've got you thinking about the next steps for the trial, you've randomized over 22,000 patients. You can relax a little bit. Tell me your thinking about what the key secondary questions might be that you could address in this population. Assuming you demonstrate safety and shorter emergency department stay, you're going to have an enormous international cohort. What are the key things you'd like to address within the trial?
I think one of the key things is really feasibility. So is it really feasible to perform the second blood draw around the 1-hour window? Is that feasible in large, busy emergency departments? I mean, our emergency department in Basel, we implemented the 1-hour algorithm years ago, and I mean, we derived and validated it. So we taught our staff how to apply it, and still we see that not in all patients it's done correctly. So feasibility, I think, is really something that I hope to investigate further and to get interesting results with this trial.
Another question. Lots of different healthcare systems, which is one of the key strengths. Are you standardizing in any way, shape or form the guidance you give on the follow-up and management of patients who are ruled out or are identified in the observed zone? Or are you allowing healthcare systems just to use whatever their local pathways are?
Yeah, so do you mean for the 3-hour algorithm at the beginning, or?
No, as part of the intervention and the standard care, once they've been triaged into these 3 groups, do you give them clear guidance as to what to do? So if a low-risk patient in Australia gets a treadmill test and a cardiology appointment 5 days later, but in Scotland they get told to head back to the fish and chip shop and don't come back.
Yeah.
How do you deal with that in the trial?
Yeah. So very, very, very good point. Thank you. So we don't address it at the moment, actually. So it's the centers perform the follow-ups. We don't have an impact on what tests are performed, for example. So this is really up to the centers. But finally, I think it reflects the different healthcare system and how they treat and continue the patient care.
Okay. Oh, one more question. Brilliant.
I have a question about what you mentioned as your feasibility issue. When I have a very busy waiting room with people with chest pain, I'm really looking forward to immediately taking the second troponin 1 hour later. What is the problem you're having? That it's taken later and it's giving you false results?
Well, yeah, the question is it really a false result? But it's the way that you understood it. So it's really about the time window, at the time point. Can it be taken around the 1 hour window? So we define it at 1 hour plus minus 20 minutes. Is this really feasible? I mean, I've worked in Basel, and it's easy because the nurses are very well trained, and they know, okay, after 1 hour I will take the next troponin. They don't even ask you. But I assume that in very, very busy and even larger emergency departments, it might be an issue. And it might really impact on the timing when the blood is taken.
In which way? That you're assuming that the troponin will be higher if they're waiting longer?
Yeah, or that it's not taken.
That it's not taken. Okay. Working in Basel, I know that we're very troponin friendly, so we're always looking forward to taking that second one and kicking them out as soon as possible.
Yeah.
So, but maybe that's not in the rest of the world.
Exactly.
Wonderful. Thank you very much, Jesper.
It's now my great pleasure to introduce to you the captain of the international football team Saturday morning, Mr. Luca Köchlin. So everyone who wants to join the international football team, please be very nice to Luca and contact him either during the break or during the dinner. In his real life, apart from being a cardiac surgeon, he is interested in cardiac troponin, and he will tell us about the direct comparison of high sensitivity cardiac troponin T versus high sensitivity cardiac troponin I in the early diagnosis of acute MI. Luca.
Dear Christian, thank you very much for the nice introduction. Dear Professor Mills, dear friends and colleagues, thank you very much for the opportunity to be a part of this fantastic meeting and to talk today about the direct comparison of high sensitivity cardiac troponin T and I in the early diagnosis of myocardial infarction. I'm very honored to be a part of this senior scientists lecture, and I was quite amused by the official invitation from the committee pointing out that senior scientists, as a rule of thumb, in contrast to masters, do not yet have gray hair or still have hair. But I think, of course, the committee is still right, since even if I'm not a statistician, I think there's a clear correlation between the age index and the amount of hair. When I look at myself, honestly, I do not even know if I have an age index, but my hair is getting lighter and lighter from year to year. But when I look at my more experienced colleagues and previous speakers, but also role models, they have, of course, a much higher age index, but also the amount of hair is getting a little bit lower. And I think in this room we all would agree that we know at least one absolute expert with an almost unmeasurable age index, but also an almost unmeasurable amount of hair. But as always, there's an exception, and I call it the Mills paradox, and I'm very eager to meet Nick later this evening to talk about secrets how to get a high age index but still keep the hair. I don't know if troponin is the solution, but I'm very happy to talk about it today, and these are my disclosures. But my most important disclosure slides are this one here. I'm very thankful for the support of the Cardiovascular Research Institute in Basel to open the doors also for a surgical resident, and for the great network for the support. So the clinical introduction of high-sensitivity cardiac troponin assays represent a major achievement not only in the early diagnosis of myocardial infarction. The current guidelines for the early diagnosis of acute MI recommend the routine measurement of one of the cardiac-specific isoforms, either cardiac troponin T or troponin I. Further, they assume that both cardiac troponin T and I concentrations reflect identical pathophysiological processes and are therefore equally effective in the detection of myocardial injury and therefore myocardial infarction. However, we also do know the differences between cardiac troponin T and I have been reported, especially regarding diurnal rhythm, prediction of all-cause or cardiovascular mortality, and, for example, release dynamics. So it seems a little bit that cardiac troponin T and I are same, but still different. So we aimed within this project to directly compare cardiac troponin T and I in the diagnostic performance for the early diagnosis of acute MI, with a special eye to the relevant subgroups of female patients, early presenters, patients with renal dysfunction, known coronary artery disease, and elderly, but also regarding the prognostic accuracy for all-cause and cardiovascular death and myocardial infarction in the future. Furthermore, to evaluate some potential clinical consequences, we aimed to directly compare troponin T and I when embedded as recommended by the ESC in the respective assay-specific algorithms. To address these questions, we used data from our international multi-center PACE trial, where we included patients presenting to the emergency department with chest discomfort. The final diagnoses were adjudicated by 2 independent cardiologists, and I want to spend a little bit of time to further highlight the adjudication process for this trial. The final diagnoses leading to the ED presentations were adjudicated according to current guidelines and the 4th universal definition of myocardial infarction using 2 sets of data. First, all available medical records obtained during clinical care up to a 90-day follow-up, so including clinical history, physical examination, results of laboratory testing and radiological findings. And second, study-specific assessments, including detailed chest pain characteristic and serial measurements of high-sensitivity cardiac troponin concentrations obtained from study blood samples. For this project, we really performed 2 totally independent adjudications. First, using as part of the study-specific assessment serial measurements of high-sensitivity cardiac troponin T concentrations, and second, using high-sensitivity cardiac troponin I architect blood concentrations. We will always report the findings for both adjudications. We call it using troponin T or troponin I for the adjudication. So this is our study cohort. We included more than 7,600 patients in this trial, of which about 1,400 patients had an adjudicated myocardial infarction. It's a typical chest pain cohort with a median age of 60 years. About 34% of the patients were female, and the same amount of patients were early presenters, meaning presenting within 3 hours after chest pain onset. Now here you can see the distribution of the final diagnosis when using troponin T for the adjudication process. And when we compare these diagnoses to the diagnosis when we use troponin I measurements as part of the adjudication process, we see some minor differences regarding the amount of patients with adjudicated myocardial infarction, which was slightly lower in patients when using troponin I for adjudication, and regarding patients with unstable angina, where the amount was a little bit higher when using troponin I for the adjudication process. Now, what about the diagnostic accuracy of high-sensitivity cardiac troponin T and I at presentation for non-ST elevation myocardial infarction? And here you can see that high-sensitivity cardiac troponin T had a very high diagnostic accuracy, regardless if we use troponin T or I as part of the adjudication process. High-sensitivity cardiac troponin I also had a very high diagnostic accuracy, which was even statistically superior when using troponin I for the adjudication process, whereas we did not see a statistically relevant difference when using troponin T for the adjudication process. Similar findings were seen in the relevant subgroups of early presenters. Again, very high diagnostic accuracy of troponin T and I. Again, significantly superior for troponin I compared to troponin T when using serial measurements of troponin I for the adjudication, whereas no difference was seen when using troponin T for the adjudication process. And these findings were consistent in all relevant subgroups: female patients, patients with renal insufficiency, patients with coronary artery disease, or elderly patients. Always a benefit for troponin I compared to troponin T when using troponin I for the adjudication process. Now, how about the prediction of all-cause death? And here you can see the area under the curve and the respective difference in the area under the curve for prediction of all-cause mortality. And what we can see here is that high-sensitivity cardiac troponin T was superior compared to troponin I after around day 75 for the prediction of all-cause mortality. And similar findings were seen for the prediction of cardiovascular mortality. Also here, a little bit later, troponin T was superior compared to troponin I after around day 90. For the prediction of future myocardial infarction, there was no difference seen for troponin T and I regarding myocardial infarction within 730 days. Now, have these findings clinical consequences? And therefore, we analyzed and directly compared the two algorithms, the assay-specific algorithms on the left side for troponin T Alexis and on the right side troponin I Architect with the assay-specific cutoffs. And what we can see here is that both assays performed excellent with a very high safety for rule-out with a high negative predictive value and a high sensitivity, and with a very high accuracy for rule-in, indicated by a high positive predictive value and a high specificity. Also, the efficacy of both algorithms was very high, with only a small amount of patients remaining in the so-called observe zone. Only 1/4 of the patients could not be triaged into rule-in or rule-out within 1 hour. What are the limitations of this project? Even if we used a rigorous methodology, with always a chance of a misclassification in a small number of patients, we cannot generalize our findings to patients with terminal kidney failure requiring dialysis, since these patients were excluded. No specific sample size calculation was performed, and therefore, even if it's one of the largest diagnostic trials ever performed on this topic, it may have been underpowered for some comparisons. And further studies evaluating other high-sensitivity cardiac troponin I assays are warranted, since we used in this study the most validated troponin I assay Architect, which is on the market. So we want to conclude that there seem to be differences between cardiac troponin T and I regarding the diagnostic accuracy for myocardial infarction. This was consistent in all relevant subgroups. Troponin I was superior to troponin T when using an adjudication based in part on serial measurements of high-sensitivity cardiac troponin I. However, the prognostic accuracy was significantly higher for troponin T regarding all-cause mortality and cardiovascular mortality. Now, the clinical consequences of these findings need to be further evaluated, since both assays performed excellent when using in the respective assay-specific algorithm as it is recommended by the ESC. And with this slide, I want to conclude, and thank you very much for your attention.
Okay, I think Andrew was first. Likely he has the same comment as I on your selection of the pictures that you show.
Thank you very much, Luca, for the advance notice of your first figure today. It was very polite of you. And a question really regarding the methodology and the implications of your outcome data. So comparing INT is obviously very challenging to do in a robust manner. One of the issues is different hospitals will have used different assays in clinical practice on which to guide investigation and treatment. Were you able to do a sensitivity analysis where you looked at the particular assay in use at that site, and whether that had any implication for whether the I or the T assay was more prognostic?
Excellent aspect. Thank you very much, Andrew, for this comment. No, we haven't so far performed the sensitivity analysis regarding this. I think one of the main strengths of this project and of this adjudication process is that we really used samples, study-specific samples, where we really had the both assays comparing. But you're right, also the clinical tests performed at the respective clinic was part of the adjudication, and we further could do the sensitivity analysis. Absolutely.
Salvatore, please.
Okay, Luca, first of all, we have a special meal for you tonight as a captain, so take care, and so that you'll be perfect tomorrow to play. No, Saturday, so we have a special meal for you. Then the question is, it seems that now you have to perform both. I'm joking, right? One in terms of adjudication, one in terms of prognosis. But I think the question would be a little bit more complex to see which is the reason that physiopathologically you think this is the question, that are implying these two different results. Do you have any explanation for that?
Yeah, thank you very much, and I think this is the big question for all of us. I think this project also opens some doors back to basic science, where we really have to find the differences, release mechanisms, reuptake mechanisms of both troponins. We have some data, and this is work in progress, of course, at the moment, that we see that troponin T seems to be a stronger signal for chronic myocardial injury, whereas troponin I can be a signal for more acute pathologies, and therefore this could be a part of the explanation why, for example, for prognosis, troponin T may be superior, while troponin I is perhaps beneficial for the diagnosis of an acute pathology. But of course, this is work in progress, and I think there are several aspects leading to these results and findings.
Just while we're getting the microphone there, can I ask? I think that's a really important point you've just raised, and I was thinking as I was reviewing your data there that may well be the explanation. It would be nice to report, in addition to the PACE diagnostic classifications of MI, unstable angina, non-cardiac chest pain, to also report the distribution of the universal definition of myocardial infarction criteria for the two assays, if you've adjudicated both. Because I think you might well be right that chronic myocardial injury with troponin T is possibly having an impact on diagnostic performance, but yet identifying a very important group of patients that have poor outcomes. So if you have the ability to look at that, it would be really interesting.
As always, excellent comment, and thank you very much.
Do you have data about the proportion of use of worldwide of troponin I or troponin T? Do you have data?
So I personally do not have this exact information, but of course here in the room plenty of persons know how the worldwide distribution of troponin T and I is. I personally do not know it. We in Basel, we use troponin T for the clinical course, but for the study examples we had both.
In terms of cost, of price, is a big difference between one and the other?
It probably depends on the country, the negotiations, I don't know.
Other questions? Yes, one more question for Luca.
Thank you so much. It was a really interesting study. In 2019 circulation, there was a paper like a troponin I is more specific for cardiac or composite cardiac mortality, but troponin T is much more non-cardiac mortality or non-cardiac cause. Is this your idea about your study? What do you think?
Yes, excellent. And of course, we discussed these findings also. I mean, it's findings from the group, from Nick Mills and the group. When I have it correctly in mind, there was also the endpoint for acute myocardial infarction and other cardiovascular events. And this can be complementary to our possible explanation that troponin I is perhaps beneficial when it's about diagnosing acute pathologies, for example, as acute myocardial infarction. However, for the prognosis of survival, troponin T may be beneficial due to the more chronic, or to the association with chronic myocardial injury, and due to a possible stronger association with age in general.
So perhaps one last question. I mean, the difference is tiny, tiny, tiny. It's significant because it's a huge data set. So we do not know whether it has clinical implication, but can you speculate, because it was only one assay that was compared, and that other assay has a higher analytical sensitivity as compared to the T assay that was used. Could this small difference be all due to the analytical advantage that this assay has compared to the other? What would you speculate?
I would suggest to do further validation studies using other troponin assays to see if this really is a potential explanation. I think, as I said, we really have to be careful when interpreting these findings, since if there is a clinical consequence, yes or no, this is one topic. The other topics are the reasons. I think there are pathophysiological reasons for it, but there also may be analytical reasons, and therefore we need to have further validation studies using other high-sensitivity cardiac troponin I assays.
Okay. Thank you, Luca. So it's my pleasure to welcome Luella Vaughan to the stage, who is going to talk to us about a very challenging time for emergency departments during the COVID pandemic. And I think despite the COVID pandemic no longer really being a pandemic, it's still extremely challenging for emergency departments. So lessons from the pandemic. Luella, thank you very much.
Right, so thank you very much, Christian, for allowing me to speak, because I think I am the only talk in which there are no biomarkers, and I do not talk about myocardial infarction. So thank you very much for allowing me to present this work. Right, so I spend most of my time working for a health policy think tank in London, and of course we've been completely obsessed by doing COVID wrap-up, and we're doing 3 linked projects which look at change in hospitals. So I've just completed a very large piece of work on organisational and operational change in smaller hospitals. We have a cohort of smaller hospitals I've been following for a long time. We're also doing a piece of work which is funded by the English Department of Health, which is going to be used to update their building regulations for the next round of hospitals. We've been promised 40 new hospitals now that we don't have a Prime Minister. Who knows whether or not that's going to happen. And we're also feeding into a piece of cross-pollinating with a piece of work which is sponsored by the EU Public Health Consortium. So the bottom is just to show you that we're actually doing this work properly. We've interviewed over 100 people. We've done a whole lot of site visits. And we're also working with infectious diseases specialists, architects and engineers on this piece of work. Right, so the first thing to say is that everybody got their planning wrong twice. So nobody's planned that the pandemic's coming, everybody's looking at the news. Nobody's planning the first time around survived contact with the enemy. So in my own hospital, and I work in a large quaternary hospital, fully kitted out, it's probably one of the better hospitals in the UK, our plan lasted 4 hours after we got our first COVID patient for what we were going to do in our emergency department, 4 hours. And that was the same in every single hospital that we spoke to. Nobody wrapped up properly after the first wave, and it was really surprising the number of hospitals that did not make major structural change to their hospitals until after the start of the second wave. And I don't think that this is just a UK problem, although there are certain issues with money in the UK. And the first wave, actually secretly lots of people quite enjoyed it once they got over being scared. It wasn't as bad as they thought. People saw how treating the patients got a lot better. They made changes, they felt more confident, and everybody was obliterated by the second wave. So I think that's a really important point to make, and so this is one of the reasons why I think this work is quite important. Right, so what were the pinch points? Well, so in the UK, it was suggested that we actually triage the patients outside the hospital. Many EDs are too small, and so the initial triage was frequently done outside, and so the patients who for COVID versus non-COVID type symptoms. And so what happened was that, so this is Leicester Hospital, so this was the most expensive ED build in the UK, and as you can see, this is the public entrance. It basically has this teeny-weeny little awning, that's it. And many hospitals had even smaller awnings than that, so basically the width of a car, and sometimes not even that. So lots of hospitals actually had ED staff outside in calgools and waterproofs trying to triage the patients. And then, as you can see, there's a tiny space here to queue. And so it's not uncommon to have queues 100 patients long outside in the cold. So lots of hospitals, to fix this, put in pods or demountables. That didn't really work either, because if you're going to properly have a pod, you've got to have electricity, water, and then you've got to have waste disposal, and that was almost impossible. So at Leicester, they did put in a pod, and they put the pod, so on that tiny bit of grass there, they lodged in a pod, and you could actually see the cable extending from outside back around, all the way around the back of the building, because of course there's nowhere where they could plug the power in. Eventually they put a generator on the back, so these are the sorts of problems. Your waiting areas were either too small to socially distance, and again patients had to wait outside and be let in, or again, this is Leicester, so great big huge waiting room. This is one of the 3 areas which run into each other, and again there's not sufficient ventilation in these sorts of places, and you can't actually separate the patients. Inability to separate internal streams. All hospitals found it impossible to segregate their flows. So my hospital is a big trauma hospital. We're supposed to be able to deal with things like nuclear disasters. So we are supposed to be able to completely internally separate our flows, and we could not. And this is completely typical, and we're a new ED. Most emergency departments only have 2 entrances, so they have a walk-in entrance, and then they have the emergency entrance. This is a problem because, again, you can't properly separate your flows with that because frequently you have patients with ambulance coming in with trauma as well as with severe COVID. And then there's usually only one internal exit which does not cross public space. If you've got other exits, they all cross public space, which again you can't do. No one has separate lifts. Again, my hospital is supposed to have separate lifts, we don't. And frequently, in lots of places, the resus was tiny, and it frequently only had one entrance to the resus. So lots of internal problems. Completely impossible to isolate the patients even within that. So most hospitals, if you had an isolation room, only had one, maybe two, many of them not actually negative or positive pressure, and you can't switch in between them. So the usual is this. So this is an Anywheresville ED. So this is actually from somewhere in the US. I couldn't find a picture, but it's nice. But as you can see, you can't put a partition in that. Can't section that off. All you can do is separate the trolleys out. And this, of course, means that you then lose space within your emergency department and your capacity dramatically falls. Oxygen, right, so this was a really difficult one. So what was really interesting is that most places thought that they could actually deliver high flow oxygen everywhere in their emergency department, and then they found they couldn't. Most couldn't even deliver 50 litres that was needed for airvo in all spaces in their resus, and they certainly couldn't deliver 15 litres everywhere throughout the department. So, and there were major problems with capacity in the hospitals as a whole. So most people don't talk about this, but pretty much every hospital we spoke to had problems with oxygen supply. Either the pressure was insufficient, they ran out, or even within the emergency department, in some newer departments, they very sensibly put the new concentrator next to the ED, but then the ED was using so much oxygen that actually the pressure dropped across the whole of the rest of the hospital and was insufficient. And this massively limited the ability to deliver treatments in the emergency department. And again, my hospital is kitted out to deliver 6,000 litres of oxygen a minute. We usually only use 10% of that, and we ran out of oxygen. Right, inability, so, gone the wrong way. No space for donning and doffing, not enough patients at the facility, so not enough loos, and then the loos are in the wrong place, so actually the patients need to cross areas or cross corridors to get to the toilets. This means that you've then got to get them commodes, that's more work for the nurses, and it also possibly exposes the nurses to an increased infection risk. Not enough staff facilities, God, where do we even begin to talk about this? So tiny, often no external ventilation, no external light. And then in the second wave particularly, hospitals realised about the levels of staff transmission, and then they were told that they couldn't use the facilities. And so people were told to have lunch in their cars. And this was psychologically devastating. I think we really underestimate this aspect of not being able to behave normally with our peers and compatriots in a time when you really need psychological support. No lockers, no facilities. Right, so what does all of this mean? So it means effective loss of floor space. So what do places do? Effective loss of floor space, outbreaks of infection, and incredibly distressed staff for all sorts of reasons. So the usual solution was to expand into a, right, so the operational choice, I forgot to take that out last night, sorry. So the operational choices were, so attempt to separate flows within the ED, usually didn't work for the reasons that we've just discussed. Most places expanded into adjacent departments. So most places it's on the ground floor, there's other outpatients nearby, there's other sorts of ward space that can be used. And so the emergency department expanded into those spaces. And that was fine, except that made running your ED more difficult, particularly in the second wave when the numbers went right up. You also need more staff to do that. And if you've got staff who are off sick, staff who are tired and distressed, actually that makes running your ED much, much more difficult. And then there are also fights coming out of the wave because, of course, the surgeons want to start seeing people so that they can start making decisions about operations. Other stuff needs to happen. And then there's lots of fighting over those spaces and what was more important. We did come across a small number of hospitals which were lucky enough to be able to run completely parallel EDs. Now, most of this was by luck because they had admissions units next door, which could just be completely repurposed. One hospital was actually in the middle of, was about to bring a brand new ED online, and so they brought it on early and just ran two. And another place that we looked at, they had actually bought a private hospital which was on the grounds of the hospital, and they used that emergency department. And that was actually incredibly effective, but again, there are issues with staff and how you manage that. So what helped? So multiple separate waiting rooms helped. So this is Airedale Hospital. When they did their refurb, they kitted out a separate waiting area. They could use that. Double doors for triage. Again, the number of places that didn't do this is quite incredible. So again, you can hold patients in here before you lead them into the main area or somewhere else into the hospital with long corridors. This is Leicester again, single ventilated cubicles. Half these, so everywhere in Leicester has these, and half of them can be switched from positive to negative pressure ventilation. But again, this was the most expensive ED refurb in UK history. Or you could build new facilities. So this is the new doctor's mess, which was built by the Army at Oban. And again, in Oban, it's like -5 during the day. So you had to eat in your Kalgoorlie, but at least there was somewhere for staff to go to get a hot meal. And so an interesting paradox. So this is Warwick Hospital. You can't, doesn't really project that well, but it's an old army barrack. So it had been used during the war. And actually it's a whole series of separate buildings, which is then connected by an internal walkway. And somewhere like this is much more adaptable than that's my hospital on the left, which once you've designed that and you've sealed it in, it has fewer problems, but you can't change anything. Whereas at Warwick, they like ripped the roof off some of their buildings and put in new ventilation systems, and you can't do that in a brand new build. So future-proofing. Every space in your hospital needs to be big enough to put a bed. It's not enough to have a trolley or a chair. It must be convertible for a bed space. Every space must be kitted out for patient care. This includes places like waiting areas, because you don't know what you're going to have to do with that space. Lots of places had space but couldn't deliver oxygen and couldn't deliver care. Multiple entrances and exits, lots of redundant space, ventilation and better facilities, particularly toilets. God, there's not enough toilets in any of these places. So I think what's really interesting is that EDs previously have been built for safety, so particularly concerns about lockdown. So if something bad happens, you have to be able to control your exits and entrances. That means you don't have very many. But if you've got a pandemic, you need the reverse. The issue with sight lines, so everything is built so you can see in a straight line. And again, that's a problem in something like a pandemic where you need to space the patients out, and actually you might need to do something a bit different. And cost. It's about £1,600 a square metre to build something new that's in the UK. All of the things I've suggested take more floor space and a whole lot more money. And staff don't like deep ED design. They don't like it being divided up. And as I said, that can make things difficult to manage. So future-proofing will require different thinking and a lot of money. And the real question is, are we brave enough to do like Singapore, which built a whole entire hospital which it doesn't use and then saved it in the pandemic? So are we going to do nothing, or are we going to spend the money and do it a bit better this time?
Brilliant talk. Not only did you not talk about troponin or myocardial infarction, but I think that was the first presentation of a qualitative program we've had at the Great Meeting, so that was really fascinating. I have a couple of questions just while people are thinking. So if you were to go back and do it again and we had another pandemic, would you recommend to your local trust or board that you do have separate hospitals for, like some centres have been able to do, or external facilities to triage those patients?
So what we did, so I think some places that we visited actually had multiple sites. And actually the issue of multiple sites was really problematic, because then you get patients, sick patients in the back of ambulances being whizzed all over the place. And given that the burden on the ambulance service is already incredibly high, that does create problems. I think if you're somewhere like London or Birmingham or Manchester, then yes, I think it does make sense. Lots of places were able to build new wards or put in new other facilities, and actually that worked extremely well.
Okay, the other question I had, just again while people are thinking, is there's a lot of interest in diagnostic testing here. One of the reflections of the pandemic was that once we had accurate, rapid diagnostic point-of-care testing, that did seem to make flow in my part of the hospital a lot easier. How did that impact life in the emergency department?
So I think that's absolutely true, but it also depended on what you were able to do in the first instance anyway. So if you're unable to separate your flows and you can't separate your patients because they're all sitting in a big area, actually, in fact, the outbreaks of infections were often worse in the second wave of the pandemic than they were in the first, even although we actually had better testing by that stage because of the problem of being unable to separate the patients. If you could separate the patients and you could test them in an hour, that's awesome, but lots of places were completely and utterly unable to do that because of reasons of internal design.
I'd be interested in Frank's perspective from the US and how US emergency departments handled this, and whether you had the same pressures as we had in the UK. Not to put you in the spot, but...
That's all right. We got eaten alive. And I'm in Houston, and you would think that it's warm enough year-round to put people out there, but it's raining, and people who are running a fever really don't need to be in 90-degree heat. So we built a bunch of tents outside, and our problem was we could make space. And apparently we have better portable toilets than Luella does, but we could even handle the toilet problem. But oxygen was a real issue, and staff, we got sacrificed because, and it wasn't doctors. We could squeeze the doctors and they'd work harder. When you run out of nurses, you're out of nurses, and that killed us. And Richard, you had some fun experience.
Detroit was very badly hit in the first wave, and we built portable tents outside and tried to triage people, and it did all of that. And staffing was a huge issue. There was tended to be enough doctors, but not nurses or techs, and that's not a good situation. The other thing that was just sort of irritating to us is that the institution had problems with staffing and stuff, so there was nowhere to go. So we're talking about building big things, but if you could have got them out, you didn't need the big things. So, it was sort of, we became the solution to many things by default because we were there. And it just made it very stressful and very difficult because of that. And sometimes you felt, all the administrators went home. You said, oh, man, that COVID problem. We said, yeah, we got them all. And so I think that when you talk about the future, it's a design of the institution, the place. It's got to be a bit of like an accordion in the ED and in the institution. Otherwise, it just doesn't work. It was a terrible situation.
So we've collected data for the whole hospital, so all the way from the front through to the back. But obviously, I've got 10 minutes for an ED audience. But you're absolutely right.
Okay, well, thank you very much for that discussion and wonderful presentation.
Now I have one very bad and one very good news to share. Which one would you like to have first? The bad one. Okay, so the bad one is Dr. Pedro Lopez Ayala, the next speaker. He selected on a short notice a slightly different title than the one that's in the program, one on which he was only able in the last 2 days to finish the analysis. The problem with that project, it includes a commercial partner, so the data still need legal clearance. And I checked my email last time, 1 minute ago, and we still have not received the legal clearance to share the data today. We hope we will receive the legal clearance to show it tomorrow. So the good news is, this is the end of the first day, 8 o'clock, dinner, 5th floor.